Good evening. This is Taxonomy Tuesday from New Jersey Mycological Association. Our members share their recent fungal finds. Um, if anyone else wants to show anything that they didn't send an email in for, if you can just put your name in the chat and we'll give you a call up. Um,
I can start it
Chris, if you,
off.
okay, good.
Oh man, me
It's only been a week since you've done this.
screen. Is it sharing? No.
Not yet.
Oh, I hit the button too many times.
Okay, now we see you.
All right. I was thinking this was Aureoboletus, but what kind? I don't know. There was a couple of these on the table. They had really nice bright pores. That's a good color for the cap too. That's a pretty good picture. It's like, you know, brown with that like reddish.
Can we see the type, please?
Yeah, I think I called at least one, not this one. I called one of these a Nixus.
Yeah, that's what was my guess too. But Enixis actually is a species group. I think there are probably four entities in there, as far as I remember from the trees that have been sent by Colin Damnauer. But I don't have a better name than that. It's definitely not Aureoporus, and it's definitely an Aureoboletus.
And it
So do
and
they
it's
have that funky witch hazel odor or that penetrating kind of a chemical scent that is quite characteristic for these things?
I didn't really notice anything like sticking out. It just kind of smelled
yeah,
like a mushroom.
Yeah,
yeah.
yeah,
And
okay.
the one
Well,
and the one that again, not this particular specimen, but I think Chris, you and I agree they were probably the same thing.
Yeah.
I didn't get that odor on the other one either.
yeah, well, now that dog-legged extension is that just a piece of dirt, or this is the actual stipe covered in dirt?
No, it goes down that way, but it was kind of like under a stick a bit.
Right.
So
You
I
know
don't
what
I
would?
don't know if that's a thing or not.
Okay. I mean, typically, like the thing that we identify as Innixis has that odor, and it has a bulbous base that terminates in a pointy root. And actually, quite a few of Aureoboletus have that kind of a bulb. Like, as a matter of fact, there's a species described from the Florida Panhandle called Aureoboletus abruptibulbus or something like that. It has a really, really bulbous base, and you can also observe a somewhat, bulbous base in Roxani, which is another species of Aureoboletus, a very distinct one. So that kind of stuff sort of permeates at least that part of the genus, because the genus itself apparently is very complicated now. I mean, there's so many species in Aureoboletus, and I've seen a paper on Aureoboletus from China recently, and that makes me think that probably we have. I would like to think more than one genus, and everybody's lumping everything into Aureoboletus with very variable morphology. I mean, just think about this. Aureoboletus betula. I mean, and compare betula with something like this, day and night difference. You would not even think they're in the same genus. Anyway, I can talk about this forever. But again, this is the best I can think of, and I have my doubts. But again, it's work in progress. We have to describe these species, we have to establish species concepts and so forth. And this is going to take time, unfortunately.
Best name for
I-
this?
So Aureoboletus is probably about as good as it gets.
Aureoboletus indexes group. Yeah.
All right.
Yeah. So did anybody save this, or just it was trashed?
I did not.
Yeah. Oh well. I guess we'll never find out.
And then this is what I was thinking was the oh man, what were we calling it?
Laconia.
Yeah.
Okay.
Elongata, maybe?
That was the other name that I wrote down on the slip.
Yeah.
I wrote Flavoconia, question mark, Elongata, question mark.
Let's see. Okay, is this the only picture, or we have more pictures? Let's see with the base. Yeah, the base is validae, and the universal veil remnants that are stuck together with dirt on the lower stipe or below the ring also are indicative of section validae. I'm not certain this is Flavoconia. It's too yellow in the cap, and it's also very white in the stipe. So maybe this is a good candidate for Elongata, which remains a cryptic species, as I told you before, with no sequences, and more importantly, no type sequence.
Interesting.
But it's in a broad Flavoconia group, I would say. But if I were there, I would have saved this collection for sure. Any more pictures of this?
Nah, that was it. It was kind of like a last minute.
Yeah. If Dave is here, he can comment on this, whether he agrees or disagrees with me. But I don't think this is Flavoconia in a strict sense.
Cool. Yeah, that's all I really had.
Hey, Chris.
Thank
Paul.
you.
Paul, you ready to go?
Thank you for
Yes, I
showing
Post in the
am.
chat.
Yes, let me share. There we go. You can see that, right?
Not yet. Now we can. I
Okay,
can.
good. So I found this one just the other day. I think this is genus Neoboletus. Found this in a grassy area under some rather mature oak trees.
Pretty.
Yeah, it is.
Yep.
Very easily marked here. In fact, on this one to the right, you can actually see my fingerprint on there. But I like to point out the stipe on these, which looks mostly yellow with what looks like some red punctee on there, because that's how it looks to me. This is just another shot, just showing a little bit better the underside. And then I did cut it in half, and it turned blue right away, which
Naturally.
not too long fades into more of a gray brown. But I don't know. Could this be Luridiformis, Neoboletus luridiformis?
No, it's not. That's a European name.
Oh,
You can
okay.
forget about all European mushrooms, boletes.
Okay.
Almost all of them, with a couple of exceptions, or I should say maybe a few exceptions.
I
But
see.
none of the Neoboletus that they have there, we have here, and vice versa, unless they're introduced, like it happens with some boletes or mushrooms in general. Most likely, this is what will eventually be known as a num nav, not a species nav, but a num nav. New name. Neoboletus subluridellus. Right now, it's still in Boletus because it hasn't been transferred. And when I was doing research on Neoboletus years ago, pioneering essentially that research, I called the species the chameleon because of how variable it was in morphology. But all the collections that were variable delivered the same sequence. So that's why I called it the mutable chameleon, and that's what it likely is. One thing that a little bit sort of surprises me are the punctae on the stipe. You said they were red; they were not brown.
To me, it looks red, but
Yeah, there's a possibility. I said, maybe it could be. Not my top proposal, but maybe second one. An undescribed species that is currently known as Neoboletus brunneosquamulosus, and that's my name. Again, established through sequencing of collections, and it doesn't match anything in the literature. So that one apparently is undescribed. I can write down the names for you in the chat. But this is below the Mason-Dixon line, right?
Yes.
Okay.
You said subluridellus, possibly on that first one?
Yeah, currently known as Boletus subluridellus, but it's obviously a Neoboletus. So, and that's an oak associate, and it's also next of kin to Validae. They are directly genetically related. There's like nothing in between. They always show up as sister taxa in all the trees for all the loci that I sequence for them. So Saproamanita is conifers, hemlock, and spruce, whereas this guy is always hardwoods, oak.
Okay. Yeah, definitely the area I found it. Very good.
Subluridellus and Neoboletus in quotation marks brunneosquamulosus.
Okay.
You can look them up on MO, those names.
Okay.
They're probably on INET as well, as people probably sequence more.
Okay. All right. Thanks very much. Let me just show one other one. And this one.
And by the way, Subluridellus is a very common species. It was described from Michigan, but it's everywhere going into southeast, Canada, and down to Louisiana, albeit it's more rare in the south than north.
I see. Here's one I think is Saproamanita theirsii. This one came up in my yard. And so I had a chance to observe it for a couple of days. I think that's what this is, a saprobic amanita that's living off of perhaps the dead roots on the grass, or maybe the dead thatch down in the grass.
I agree with the species name, but I'm strongly against genus Saproamanita. Amanita is monophyletic. And any attempts, and I know who is doing this, I know whose name this is, I completely disagree with that treatment.
I see.
There's going to be people on both sides, but all the recent papers in Amanita kept the genus monophyletic. So all these saprobic amanitas that are now in subgenus Lepidella, section Lepidella, are still part of Amanita.
Okay.
But yeah, I believe this is a good idea. I've never seen these in the wild.
Yeah, I've noticed. I don't think you really have any. I don't see any sightings in New Jersey. Certainly not on iNaturalist. At least there's a few, but they don't look particularly legitimate to me. There's some in southern Pennsylvania, but
right. That's always a problem. Misidentifications, and I don't go to iNat often, but when I do, I mean, I see some really horror stories.
Yes,
bad observations that are clearly misidentified, and nobody's doing anything about them. It's just like, yeah. At least when you're on MO, you're going to be facing some people who actually know their stuff.
Okay. Yeah, if I'm correct, this one first showed up in the 1950s in Texas, or at least first documented there, and is it possibly spreading its way north, and from there, I don't know, maybe you'll get it in New Jersey eventually.
Yeah, just earlier this month, I found a bolete, which is probably the first collection of that species in New Jersey. And I found this in a spot that, for me, was the most frequented spot in the last 20 years. And I've never seen a species growing in there. 20 years. Visiting that spot a few times a year. Every year. So go figure.
Yeah.
But, another comment that these Saproamanitas are very ancient. They are extant species from an early divergent lineage in the genus, because they are saprobic, and the mycorrhizal relationships have evolved much later on. And mycorrhizal amanitas have lost their genes that code for the enzymes that break down the organic matter in the soil. So you're looking at a relic, one of them.
Interesting. Yeah. All right. I wonder if they still have that name in iNaturalist. It would just otherwise be Amanita theirsii.
Yeah, Amanita theirsii.
Okay. Well, I think I'll just stop
I think
there.
it's Scott Redhead's name. It's his idea to separate Amanita, or split Amanita into two genera, one for mycorrhizal Amanitas and one for saprobic Amanitas, like this one.
I see. All right.
That's
Thank you
a
very
really cool specimen, by the way.
Oh, great
Well,
shape!
thanks.
Very fresh, very photogenic.
Yeah, it's interesting. I had a little bit of a doubt because the literature that I was seeing, let's say like on Mushroom Expert, they talk about the stem being sort of topping out at about 7.5 inches, and this one is closer to 9 or
Oh
over
wow!
9 inches.
I thought it was smaller than this. It certainly did not make that impression on me.
Could be 10 inches, something like that. But yeah, I think some of the literature is sort of shortchanges it.
So this specimen actually was 10 inches tall?
Certainly from bottom to top. I mean, if I were to just extract the cap, which wouldn't add too much to it, yeah, it was about at least 9.
Yeah, that's remarkable. That's quite large. And this is still like an immature specimen.
Yeah. So that's interesting. Thank you, Igor.
My pleasure.
Thank you, Paul. Next up in the chat is Svetlana. Are you ready, Svetlana?
Are you muted?
No, she's not muted. I see her now, but we can't hear her. Svetlana, can you hear us? Okay. Yeah, but we still can't hear you.
Well, even though her microphone appears to be unmuted, the icon is different. There's no sound, so it must be something to do with the computer, not the
Right. It's on her side.
yeah, not the website. Yeah, okay.
All right, so we see your first picture, Svetlana. Red port bolete.
Yeah. Okay. Yeah, I think this is subluridellus, just the standard variety.
That's your chameleon, right?
Yeah, it's the chameleon. Yeah, that is classic look. Yeah.
What is the genus that you just mentioned?
Well, it's Neoboletus.
Oh, it's Neoboletus. Okay.
Yeah, it will be
understand yet.
Neoboletus. It's a red port bolete.
Okay.
Although there's more than one genus of red port boletes, but this one is the most prolific one, at least in Asia. So yeah, growing with oaks, and it's going to bloom everywhere as they always do. Nice red pores.
And Igor, we don't have to go back to the photos, but that can have yellow pores, right?
Occasionally, when young, but typically they present themselves as red, and then they fade to more kind of orange. And there's always frequently like a band of yellow pores around the rim of the cap. So you cannot see it here because it's probably still young. But as they get older and the cap expands, it's not a constant trait, and it's not a trait that should be used for any species identification. But like I said, I've seen in this species often enough, there's going to be a yellow rim of pores right where the cap cuticle begins.
Cool. Thank you.
What species did you mention, if any?
Subluridellus.
I don't know how to spell that one.
Yeah, lurid. Subluridellus. It's already in the chat.
It's already in the chat. It's the one
Together
we were just.
I don't see it coming up on my slide.
Go
Okay.
back a bit.
Yeah, this species is typically presented with a stipe. It has a dense coating of red squamules over the yellow background. Albeit, I've seen collections that were all yellow except for the pores, and the stipe was covered with yellow squamules, no red. And another trait that's very common for the species that's going to stain burgundy, like a vinaceous, like a Bordeaux wine on the bottom of the stipe. That's where the hairs come up, like in Phylloporus. So that's also a constant trait for the species.
Next one.
Yeah, here we have a fruiting, or maybe multiple fruitings of a gilt bolete of some kind. And I see the caps are sort of darker red-brown, this rich color. The question is the mycelium at the base of this type yellow, or is it white? And that's going to basically split things up between the rhodoxanthus group and the leucomycelinus group. The question is whether all the white mycelium species are collated together and separate from the yellow ones, or there's always going to be an intermixing, and this trait is then not constant. It's of little taxonomic and phylogenetic value. Yeah. I don't know about you guys. I cannot really say whether this is yellow or white.
Yeah, I would agree.
My
with that.
eye looks yellow.
Yeah, and to my eye, the one on the bottom left looks kind of white, but aren't we looking more for? Aren't there
I think
rhizomorphs?
The one on the left could be a different species.
Yeah.
Look at the cap color.
Right.
It's more brown than red. The other one is really this kind of a maroon color, very rich. So we possibly have more than one species. The larger grouping on the right, I think, is one species, and the two fruit bodies on the left is another one, unless they were all found in the same spot.
If this was the ones that we were looking at from Shark River up at Green Turtle, people were finding them. They were very red. I mean, is that a matter of them being young, or
Possibly. It's interesting that Phylloporus in the United States and Eastern United States is a small genus, but we still probably have maybe up to, I would say, I don't know, at least 6 species genetically, and some of them are quite distinct. But the ones that look like rhodoxanthus, I think I'm aware of at least 2 species. And you can actually go back to the papers from 2011 and 10 and then about 10 to 12 years ago by different authors, including Roy Holling, where they do the phylogeny, but since they never sequenced the type collections, the names that they propose for certain clades, is very questionable at best. And if you really look into individual species, especially rhodoxanthus, how that was described and how the type was assigned to that name, you would begin to doubt even more that we have the right concept and the right species. So with that being said, I just leave Phylloporus alone. We can, of course, sequence, but we'll never get a name because most of the species in this genus present in this country, I would say in the eastern half of this country, are undescribed as far as I'm concerned.
All right, Svetlana, do you have another one? And I put a phone number in the chat that you may be able to call in as per Lila's suggestion. I haven't tried it recently, but hopefully that might work.
Okay. All right. Anybody wants to, before I start talking away, anybody wants to take a stab at this one?
What kind of staining?
Looks like
What kind of
that corrugated cap Leccinum. What's that thing called? I forget what it's called. Looks like it's got the slightly yellow pores, so I would rule out.
There's a name by Beck, rugosiceps.
Yeah.
But
I think that's what that is.
Yeah, I would say this is not Leccinum in a strict sense, but it is Leccinum according to Kuo and Ortiz as of their 2020 paper, which I think instead of trying to separate things, they just crunch them into one big heterogeneous genus.
So you think this belongs in Leccinellum or Hemilecccinum?
Well, Leccinellum is polyphyletic, so you have to define what Leccinellum is in a strict sense, and then you have to start creating. If you want to go with Leccinum, you have to do the taxonomy in terms of sections and subsections. If you are of the opinion that we have multiple genera, then you have to create new genera for these clades.
I would guess that Quote just didn't want to do that.
Quo did not want. I mean, their arguments were interesting, but I think there's a slippery slope there element.
Right.
And
yes,
and either
that's what I'm thinking.
Yeah, either way, they didn't want to deal with the complex taxonomy. They just said, "Oh, we're going to sequence these 3 or 4 genes, and looks like our statistical model gives us 100% support for this big mess of Leccinum, and therefore we're going to declare it as Leccinum because it's easier. It makes more taxonomic sense to lump everything into one genus as opposed to breaking them up into different genera." And that's what actually phylogeny or sequencing has been doing. We've been actually expanding our list of genera, not collapsing them into fewer entities.
Right. And in some cases, for a mushroom enthusiast who wants to go out into the field and have an idea of what they're seeing, it makes sense because often these split off genera include species that correlate morphologically pretty well.
Yeah, they're Leccinioid fungi. And I know it's very tempting to lump everything into the same genus, but this has been done before, and we're unraveling that mess that was left to us by our morphological forefathers that called everything Boletus, for instance, for a very, very, very long time, into the 80s and 90s, I would say.
Right.
At least, maybe even to early 2000s, before the advent of DNA sequencing on mass scale.
So you mean from the last century, the 80s and the 90s?
Yeah.
How old is genus Leccinum?
Leccinum gray? Old,
yeah.
But even that genus is controversial the way it was named. I remember a big discussion with somebody on MO about this, maybe Joseph Cohen,
Let's not get
But
sidetracked.
but the bottom line is that you want to call this Leccinum rugosiceps. I'm not gonna oppose this. Everybody is entitled to an opinion, whether it's just personal preference or something that's more grounded in science.
Well, that was the name I was trying to think of at any rate. Thanks.
Mm-hmm.
What was that growing with? Is that a conifer associate or?
Hardwoods, oak.
Oh, okay. Thanks.
Yeah.
Thank you.
Yeah. If you really want to see about those types of Leccinum, search the name Leccinum luteum on MO and actually check out the sequence collections, because now we have a type sequence, and you'll see that it's one of those things that look like that, like rugosiceps, not the true Leccinum in the sense of vulpinum and snellii or scabrum. Even though scabrum I think is probably not here, or it's rare. But yeah, okay. So different bolete here. I think I know what it is.
Is this Hemilecccinum subglobropus?
It's not
subglabropes. That's what I was going to say.
No, this is not subglabropes.
It's not subglabropes. Huh?
No.
The stock looks a little smooth, I guess.
Yeah, this is Leccinum aenigmaticum. This is a provisional name
it's
by me.
Enigma.
Yeah, because it stumped us for a very long time. It stymied us for a very long time because its LSU and ITS sequences did not place it into Leccinum. Only TEF1 gene, when it got sequenced, placed this into Leccinum. Hemilecccinum.
Hemilecccinum.
Yeah. So this is the enigmatic Hemilecccinum, and it is a widely distributed species. We have collections from Louisiana, we have collections from New Jersey, and pretty much everywhere in between. Very easy to ID morphologically from pictures. Oh yeah, that's a classic. That's a classic enigmaticum with that bloom on the top.
So is the stalk smooth on these like it appears in the photos?
The second photo, Dave.
It has the squamules. It has the scruffiness.
It has the squamules. Okay.
Yeah.
Yeah, because I didn't see it at first.
If you can zoom in at the top of the stipe at all, Svetlana, I think you might be able to make it out in this photo.
I'm going to type in the name so you can search this on MO or maybe on INAT if they use it there.
It doesn't look as ornamented as subglabropes, though. That has like little ray. That has almost like scabers.
Yeah, this has scabers too, but they're not as prominent. You can actually see this type kind of breaks up.
I see it now. They're kind of flat.
Or scabers. Yeah,
they're
more oppressed. Yeah, they're more oppressed. They're less scruffy.
Right. It's a nice picture, actually. You can see the pores really well. You can see some of the detail on this stipe. Very nice. Svetlana, did you get these in the Poconos last weekend or somewhere else?
Dave, her mic's not working.
Oh, no.
Type in the chat.
Yeah, type in the chat.
She would have to stop sharing, I think.
Oh.
You can type it in later. I know she was going to spend some time in the Poconos.
Later, yeah.
Yeah, well, like I said, this is a widespread species, and we have examples of this. Actually, we found this at Smithville a couple of years ago. I had it sequenced, even though I knew what it was. I still had it sequenced. It's not a very common species, but you can come across it, once in a while.
I don't think I've ever found it. I don't believe I've ever found this. Or if I did, I would have called it subglabropes, but it would have bothered me that the stock doesn't look quite right.
Yeah, subglabropes has a much paler cap color. It's more like yellow brown or kind of warm caramel brown, where this guy starts off really, really dark, and then it gets reddish brown as it ages. Of course, there's always
When
morphological plasticity in any species, and you're going to see deviations from quote unquote the norm. It's a Gaussian curve. You're gonna have exceptions on either end, and then you have most species following a more recognizable trait in the middle. But one thing for sure, in New Jersey, Hemilecccinum subluridellus is a very rare species, and you would only find this in northern New Jersey growing with birch in mixed woods with hemlock.
Her photos are from Huntington County, New Jersey.
This one is from Huntington County. Okay. But subluridellus, we found this at Meadowwoods in Morris County, and another collection came from Stokes, in my memory. So right now I'm searching for the species on ML. I just want to see the distribution range to give you a more accurate picture of how widely distributed this is.
Well, what do we think about these Tylopilus?
Are those two different species there?
So
I would say yeah.
So closing on yeah, the ones on the left are plumbeoviolaceus, I think, and the ones on the right are varietatibus. We have to zoom in to see the details, but they are definitely two different species. The one on the left is bitter. The one on the right should be mild tasting.
Oh, do people eat that one? Varietatibus?
Varietatibus.
Yeah, varietatibus.
I think somebody's been eating them. Svetlana. They are the Svetlana McCoy Rusanova.
Okay.
So she ate them, I think, and to my knowledge. So closing off on aenigmaticum, collections from Connecticut, west to Ohio, down to Louisiana and Mississippi in the south. Virginia, Maryland, none in Pennsylvania, and none in New York so far.
That yellowing at the base is interesting.
I think it's an artifact of the iPhone photo software.
Okay.
Maybe.
What are the ones on the left?
Plumbeoviolaceus, naturally.
Oh, okay. All right. Yeah. It might be some environmental factor as well. The yellow.
Yeah, could be staining from, I don't know, something else. Could be just an artifact. But these guys. Can you zoom in on the ones on the right again? I want to see if they're reticulated. Maybe not. If that's the case, that's badiceps.
I was wondering. Yeah.
Yeah, now I can see the lack of reticulation and some of the beveling.
It looks like the cap is beveled.
Chamfered edge on the bigger fruiting body, and often these are going to have multiple colors. They start off as dark burgundy, purple, and then they transition into this mix of warm brown and burgundy together, and so that's unusual for badiceps. I don't see any articulation, so I don't think this is Variobrunneus.
But just
it looks like the caps are beveled on the margin. Look.
Yes, I can see the beveled on the bigger fruiting body, and not so much on the one on the bottom.
Yeah, on the bigger fruit body on the right, you can see it's got like that little angle.
Yes, and it's not going to be bitter. Badiceps is one of the best edibles in genus Tylopilus. That and now that Alboater has been moved out of Tylopilus, that's delicious too. So we're left with Tylopilus badiceps as the most delicious Tylopilus.
Yep, Ferruginous is okay, but it's not great, but it's okay.
We don't know what Ferruginous is, Dave.
Oh, Oh, you mean like nobody knows what it is, or you just don't get it in New Jersey?
No, nobody knows what this is, and I'm actually also skeptical about
Oh,
badiceps.
good, because I just sampled one for sequencing.
Yeah, but there's no type sequence, and you have to go to
Well,
the
but at least
description.
we'll see what it matches.
Is
I mean,
plum
it's just going to show you that the species that we call badiceps that has a unique sequence is going to be different.
Well, okay. Well, what the thing I found I think is ferruginous.
Well, my question remains: how certain can you be if we don't know what ferruginous is?
Well, no, well you can't, but you still can group together sequences that
are
have
different?
similar enough that are similar enough and say they're probably the same. I mean, yeah,
Well,
using the
you
name
can
is
group
dubious. I
sequences.
understand that,
Yeah, you can group
but in terms
sequences.
of there being a certain taxon, and
Yeah.
you want to try to understand it, how do the sequence? How does the DNA group together? I mean, that's one of the first things in today's world. That's one of the first things you want to try to find out.
Right, correct. You're drawing a parallel between sequences and the likeness and appearance, and you can say, okay, we're getting a species concept, but is it again the species that was described 120 years ago?
Oh well, maybe not. We might need a new name, but whatever,
But
whatever.
yeah, so
That
basically, the modern concept of badiceps, which I think is this, um, yeah, it's supported by sequences, certainly, and
so have they sequenced the type of badiceps?
no, no, no.
Okay,
That's the problem. That Tylopilus
okay.
is still pretty much in terra incognita.
So most of the Tylopilus are too old, I guess.
Most of the Tylopilus are too old.
Are they like PEX things?
Yeah, Pack or
Yeah.
Frost. I have to look this up quickly.
Okay.
But yeah, it's mild and delicious and very tasty.
Is Tylopilus mainly an eastern half of North America thing?
Yes,
Do they have Tylopilus in Europe?
only one, the bitter one.
Oh,
Thalius.
that's why my friend Boris from Moscow called it the Polish mushroom. Because
No, no, no,
they knew
no,
about it even in Russia.
no, no, no. He's wrong. The Polish mushroom is Imlaria badia.
Are you sure?
Absolutely.
Because I showed him Tilapilus phallius and he laughed at it.
No,
And he
no,
called it the
he's wrong.
Polish mushroom. But this is a common name. It might vary regionally.
No, Polish mushroom, or in Russian it's going to be pansky grip. The other name is polsky grip. That's definitely Imlaria badia.
Oh, well, that's
Yeah.
refreshing for me to know. At least they're not calling this practical joke mushroom the Polish mushroom.
That doesn't fit the narrative, though.
That's sort of what I'm saying. Yeah, this yeah,
Yeah,
Mary
I
Patty is
mean,
actually a pretty good edible.
I don't think you want to name a nasty tasting bitter bolete after a country. That would not be a very good practice.
Well, no, but different countries make up jokes about the other countries. I mean, that's like
Yeah,
just
but
a
that's not the
classic
kind of
sort of
joke that's
cultural
funny.
thing.
That's kind of joke that's demeaning and insulting.
Yeah, well, that's people think that stuff is funny,
Well, in the 1970s, we did, but I guess we've changed. We no longer find these ethnic joke books in bookstores, so I think it's passé. We've
By
moved
the
on.
way, Boletus badiceps, later known as Tylopilus badiceps, is a PEC species from 1900.
Wow,
And
neat.
I encourage you to go to Index Fungorum and click on the protolog link to read the description and then ask yourself a question. What we're seeing is that species or not?
Well, it's hard to tell from any of these PEC descriptions,
And
which
that's
are
my point
typically
exactly.
like 2 sentences.
That's my point exactly. You don't know to certainty.
Okay, well, thank you, Svetlana, for sharing. I think that's your 5. Next up is Jason, if you're ready.
Sure. All right. So I shared this one a couple weeks ago.
Yeah.
Probably something in Otidea, but at the time I had just gotten back from the field. I didn't have a chance to get spores out of it. Um, so I shared these photos, but here are the spores.
Ooh.
And you can see here how on a lot of these, I guess this is a paraphysi, or you can see the paraphyses are kind of hooked at the end, which appears to be an important character in the keys.
Those
So,
are asci that we're seeing right now.
Right? Yeah. All of them, even the empty one.
Yeah,
Yeah.
those are asci we're seeing right now.
Right.
The paraphyses are going to probably be a lot thinner. See the spore. If there's spores inside it, it's definitely an ascus.
Right.
And the one on the left is also an ascus. It's just
Empty.
it's expelled its spores probably, or it's
Okay.
sterile. Sometimes they're sterile.
So I've been going off of this key here, from fungi temperate Europe. And I highlighted all of the spore lengths, and you can see, a lot of these are, this is a 400, so each of these tick marks should be 2.5 microns. So a lot of these spores are coming out to be upwards of 20. One, 2, 3, 4, 5, 6, 7, 8. And that really narrows it down. All these ones that are red appear to have spores that are too short, and all the ones that are green are the ones that are larger. So you can see here the paraphyses that I guess I didn't get any photos of. You see some of them are, they curl all the way around. These are all straight. So I called it cantherella. That seems to be the one that's the best fit. But it's I got to the point in the key where I had to say, is the outside buff yellow to apricot orange, or is the outside more or less buff honey yellow, or more or less brown to dark bluish gray? See if we...
Hey, Jason, was it just the two microscopy photos, or did you have a third?
No, one is just a zoomed in photo. There's this one, and then this one I just zoomed on my phone, so it's the same as this one.
But that other photo, there was, it doesn't seem to be as great. There was another one, right?
Yeah. Let's
This
see
one?
what this shows. Those are all asci, I think.
They are, but
Yeah.
there are some paraphyses out of focus on the actual clump. Yeah, there.
Oh.
Like that one that is they extend above the asci, and you can't really make anything out. Yep, right there.
Yeah, but
you can't
either
really
way, the
make any.
ones that have straight, it's just these 4 species that have straight paraphyses, and the spores are all too short. So,
Gotcha. Yeah.
um,
Jason, I noticed that's a key to European fungi. So does the key say whether or not it's in North America or whatever?
yeah, so I mean, a handful of these species are on Myco, Quebec. So I think, like with a lot of these
Yeah.
species, we all we have are European names.
Right. No, I was just curious, like, if there's any chance it wasn't here. But yeah, you're a very thorough person, so you probably
Yeah, I mean, it
check
could
that
certainly be a
out.
species not listed here. There are a handful, but a lot of species are also dark. The fruiting body itself is dark, which is how I like ruled this guy out, and I believe this guy. So, um, yeah, I mean, I thought that the second couplet here kind of fit better. The not the dark bluish gray, but it did look a little, less than buff. But I don't know.
Yeah, yeah. I mean, I wouldn't disagree with that.
I think this at least rules out the brown and the yellow ones, which seem to be more common in my area.
Right.
So
The microscopy is beautiful. It's so clear and nice.
Thanks. All right, so then I've got a bolete for Igor. Another reddish to
Okay,
orange cord.
that's an
This
easy
one
one.
doesn't have the scabers on this state.
Yeah, that's another non-prof that's going to wind up a new species. This is Neoboletus varietatibus or varietatibus, depending on where you put the accent on what syllable. But the color scheme of the cap alone is enough to identify this bolete from the top. You don't even have to look at anything else. It starts off
Bright red.
as dark brown with olivaceous brown areas, and then as it matures, it develops these dark red tones intermingled with oranges and yellows. So varietatibus in Latin actually means multicolored. Back in the day when I used the Google Translate from English to Latin, that's the name that it gave me. So it's a very attractive Neoboletus, and the interesting part about it that it's completely unrelated to anything from the genus in this country. Its closest relatives are all in China and Southeast Asia, like Thailand, for instance.
On the edge of the pore surface there's like a distinct yellow. Was it one color and then changed to the other, or?
Yeah, I know you get that with mostly, like I said, I saw this in subluridellus, but you can get it apparently in other species as well. It's not a trait that should be um, uh, treated as
Relied upon.
characteristic of any particular
yeah,
taxon.
you see it in subluridellus also.
Yeah, well, Dave sees subluridellus a lot. I don't, so I quickly forget.
Yeah, it's pretty common in Neoboletus as far
Yeah,
as I can see.
but this critter
That's the
again
flash.
really has a unique looking cap. And when you see something like this, you can exclude everything else in the genus. It's only this species.
Yeah,
It's
that
Igor, can you
doesn't.
put the epithet in the chat?
Yes.
Is it variapes?
varietatibus, not varipes. No, varipes is from a different opera.
Variatibus?
Neoboletus, like this.
Okay.
And again, MO is the place to see them all. Some states that are very common. I think it tends to be the epicenter of the species tends to be like in Virginia, which is true probably for many other boletes as well. But it's distributed as far north as Connecticut, as far as I know. But interestingly, it's not present in Michigan and any of those Midwestern northern states like Minnesota, Michigan, and so forth. It's absent from the north,
I
but it's in the
think
south.
I think I remember finding this. I think I remember finding this once here in my area, and I believe that you identified it, Igor. So that would be the only
Possibly.
time.
Like I said, MO is the place to look up dozens upon dozens of, the species, observations of the species. Very attractive, has a unique phylogenetic profile. I think it's my favorite Neoboletus as far as I know, because it's easy to ID and because of its provenance.
And you know what? I will second Igor on this suggestion that MO is the place to look for observations and discussions about boletes. I think they've got INET pretty much. They've upped the ante on them with this particular type of mushroom. I think you're going to find more stuff on INET about Amanitas, for instance, but boletes, the best bolete people are on MO, really, honestly.
Yeah, I would recommend looking into the observations of Eric Jones, who is active on MO. He's from Georgia. He's a PhD student in mycology, but he's not doing, his project is not about alpha taxonomy. You know, it's more ecological, I think. But his avocation or his hobby are boletes, and he has his own little kind of a well, it's a website, but it's more like a kind of a not even a chat, like a journal, pretty much. It's called Bolletivore on Weebly, and I'm putting a link here. He's very eloquent. He's a good writer, and you can read about various boletes that he finds in Georgia, and many of them we find here in New Jersey. Not all of them, but some. And there are a couple of mistakes in there when it comes to pictures. I think he made mistakes. I have to contact him and let him know he should replace some of the pictures with the proper ones or the captions. But you'll derive some pleasure from reading those accounts, because this is something that I could have probably written myself, but I never came around doing that. And he put it very nicely into these nice little articles.
Cool.
Thank you for sharing that. I look forward to checking it out.
So one more time about this bolete. If you see your red port bolete that starts off as dark brown with olivaceous green flushes intermixing, and then seeing these deep, red maroon tones with yellows and oranges splattered everywhere, this is your Neoboletus varietatibus. There's nothing that looks like this remotely anywhere in the world.
Looks like somebody smeared jelly on top of the cap.
Yeah, really beautiful species. And this type is going to be yellow flushed with red, like it is the case for many Neoboletus. And you'll probably even see some reticulation in the upper, I would say, 10% of this type occasionally. It can be reticulated. This is just at the apex, but I've seen specimens where reticulation was maybe like at least half an inch, maybe even more down the stipe. Very delicate reticulation.
Cool. All right, and then next I've got three Hydnellums that I found up in North Jersey, all within 10 feet of one another. So this is the first one. I'm looking for confirmations on these species names, but here's the first one, obvious zonation on top. Here's the teeth on the underside. So I called this one concrescens.
I would concur.
Cool. You compare that one with this guy. Obviously,
what kind of habitat? Oh, nice.
This is a oak-dominated wooded slope. Here's the underside. You see, it's kind of wrinkled or crumply. It's got this kind of waviness to the cross section. So I called this one spongiosipes.
What is the stipe squishy?
Yeah, if you think you have spongiosipes, you just squeeze the bottom part of the stipe, and if water comes out, then you're probably right.
I guess I gotta find the photos and add it to this, but I also did some chemical tests according to mushroom experts. So the cap was K plus dark green, and then ammonia turned to light green. But I don't know how as far as I know, all the Hydnellums are like that.
So to my eye, is this? I mean, it looks like it's staining brown, but does this have any hints of blue in the field?
What did I remember? I mean, I could see a little bit of blue like here, gray blue.
Yeah.
Why
Did
would
you
that
cross?
point to a different species?
Well, what is it? Cerulescens?
I forget the name,
Oh,
but I know what you're talking about. It has
Yes, ceruleans.
CAER in it.
yeah.
Yeah. I think
I know
that's
these were.
really more blue, like
It's
more
ceruleus, I think.
indigo.
Yes, Cerulescens.
Like,
Yeah, we've seen this blue. I think at Bass River, Jason, you were there a couple of years ago, maybe.
Yeah, these were. I mean, I gotta have other photos of these that I'll have to add to this observation. But there were quite a few of these, and they were all about this size. I mean, you can see, the width of my finger here. These are just, 1 or 2 inches across. My understanding was Cerulescens was a little bit more sizable, comparable to something maybe not this big, but
I'm gonna put the. I'm on Micro Quebec. I'm just gonna put the link in the chat to what they're calling ceruleum.
right, cerulean.
Looks like the cross section is where you get that mostly.
Oh yeah, this one looks a little bit, when it's younger, this one looks kind of fuzzy.
Did you section any of these, Jason?
No.
Okay, yeah, Susan Hopkins has taught me to section these to look at what the context is and the zonating aspect. That's
Okay, I'll have to do
supposed to
that.
be helpful.
Yeah, this one,
was
This
this
the
doesn't.
thing
also
about,
growing with oaks or was it conifers?
yeah,
It was.
no, it was all hardwoods.
I think the ceruleum. I mean, I'm not sure, but I found it with conifers.
Yeah, a lot of these have pine needles with it, and it's also got, um, it frequently has teeth that kind of partially run
Yes.
down the
Yeah.
stipe.
Yeah.
This one, it was pretty abruptly, it didn't extend down. It was pretty much just on the underside of the cap.
But you didn't squeeze it, huh? You don't know.
No,
You're not sure. Do you still have it?
I think I do. Might still be in my truck, but it's probably super dry.
Your truck, I bet, smells like my car.
Yeah.
Something for Elizabeth to test as a dyer. It might be interesting.
Yeah.
If it's if it's spongiosipes, she'll be able to tell from the color, but if it's something else, it might be a different color.
There you go.
And then rounding out Toothy Tuesday here. This was also within 10 feet of the other two, and this one obviously has the full stipe. So
This one looks more Sarcodon to my eye.
Yeah, I think so.
Sarcodon.
Sarcodon.
Or pseudo Sarcodon.
Oh, I don't even know those.
Aren't these things mostly have been moved out of Sarcodon into Hydnellum?
I think so.
That's what I
Yeah,
thought.
a lot of them have been moved.
So that's the one that has that bluish gray staining on the bottom, right?
The green.
Yeah, well, you call it green, I call it aquamarine, whatever. But you get the picture. So it's not under woodii, and it's not imbricatum, right? So it has to be scabrosum.
Well, let's have a look at the cap.
Yeah, that would be good to see. Oh,
I
no more
had
pictures.
I got photos of the cap though. I don't know why these aren't. Maybe I didn't take one. No, I do. I have it right here. Just didn't add them all to iNet.
You can just describe it. We'll believe you. Is it
Or
It's
scaly?
put it
Scaly?
in the chat.
yes, it's scaly. It's brown. Scales are more abundant towards the center. So yeah, it looks not as scaly as this. Maybe more like this. Yeah, so
Well, it's
that's
one of the mushrooms formerly known as Sarcodon. And it might now
Well,
be in a different genus.
it would be helpful to dig out observations that have sequences because there was that paper that basically transferred a lot of sarcodons into Hydnellum from a few years ago, maybe 2021, something like that. I think that paper was by European, Swedish maybe mycologists, something like that, or Danish, I forget. And I may have reference that reference on the mouse somewhere, but they're the ones who actually studied that genus very thoroughly, and they probably dug out type collections or proposed epitypes or neotypes for these. So they're reference sequences for these taxa, and we can probably say with a lot less ambiguity now what these things are. So if you can find validated by sequencing collections of Hydnellum scabrosum and then compare this to yours, maybe you'll get a more definitive answer.
Oh, wow. That's
For
not
instance,
that scary.
on MO, you can do a search, a more elaborate search that allows you to include only things that have been sequenced or the ones that have specimens or both, specimens and sequenced. And it's only going to give you observations that are supported by DNA, not everything else that could be, in error.
yeah, cool. Thanks, everybody.
Thank you, Jason.
Because I'm curious whether it's a single species that has that greenish blue staining, or it only happens in just one species.
I could go.
Okay, Shihan was first in the emails, though.
Oh, okay.
Um,
Oh, I
Shihan.
thought we were still doing sharing from our computer.
Oh, that's right. That's right. Yes, go ahead.
Okay.
I forgot. You're not an email person again. How quickly I forget. Like 10 minutes ago.
So I have some similar stuff to what other people showed already. There we go. I believe this is Tylopilus badiceps. I would call that a beveled margin. Would others? Yes.
Yes, and not only is beveled, it's also this deep sort of reddish purple color, which is typical of the species in our modern understanding of that taxon. So yeah, I think this is badiceps.
All right, cool. And unfortunately, I wasn't able to do any microscopy this week.
It wouldn't help you with that. I don't think so.
It wouldn't have helped anyways. Yeah, good. And then last year in the same, oh wow, wait, now this has changed. In the same location, I found what I was calling Atkinsonii. Okay, okay.
I just looked at the observations and gave them names or support your proposals.
Oh,
Yeah.
did you change this to Veripes then?
Yeah, I changed this to Veripes.
Oh, okay. This is a porcini, right?
Yeah, this is a porcini, and I think this is the true Veripes. As I wrote in the proposal, I think this is the true Veripes, as opposed to all the other taxa of oak-associated or hardwood-associated porcini that have been lumped into that name by many people from decades ago.
Yep, because these look like the thing that we used to call nobilismus.
Correct.
Oh, is that name no longer?
No, it's retired. This is
Oh,
Veripes. The type collections of all the porcinis from this country have been sequenced. Like all the pet collections have been sequenced, and now we know what these things are genetically, and we can match our modern collections to the type, through ITS.
huh. Well, as young as they are, you can see how like already they're totally getting eaten. But I did collect a lot of them, and I hope to eat them. So I'm excited.
Well, save one specimen for sequencing just in case.
Oh, okay. I would love to. And I know this isn't exactly why we're here, but something I was hoping to talk about tonight, or if anyone had a good resource for home herbarium best practices for a home herbarium, but like at the simplest, most basic level, low budget, but
Yeah,
um
well, it's going to be low budget because that's your home. And Dave and I can both tell you our experiences with home herbaria. The most important part is to have enough space. If you are
okay.
collecting hundreds of specimens, if
Okay,
you are
so
just a casual collector, then it's probably less crucial. But
let me ask you if my understanding is correct. Shoe boxes with Ziploc bags labeled well with dried specimens, maybe some the packets that keep moisture away. Is that about it?
yeah, I mean, you can buy these small silicon
Right.
packets and put them into your Ziploc bags, and that certainly helps. But yeah, pretty much, yeah, shoe boxes. That's what I have. I have shoe boxes full of Ziploc bags with dried specimens, and I used to write with indelible ink on the bag, but I found out later that writing can still fade away, or something else might happen and it gets
So
erased. And the best practice is to buy these small index cards and then write
yeah.
on them in good ink, and that's going to last for many, many, many years.
However, the Rutgers herbarium required to have ID tags in the herbarium that are acid free.
Correct. You can buy, and I've done this for my NYBG deposits, years ago, from an EMF in 2018, where I had to design my own label that fit their parameters and then print everything in acid-free paper containing rags, basically like your banknotes, same kind of paper, with watermarks and stuff like that. So yeah, you can do that. You can go as far as doing this, the most expensive way. But that way, you're going to ensure that the ink is not going to be lost. It's not going to get oxidized. And,
okay. So that requirement is only based on the ink getting lost. It's not based on sort of like contamination of the specimen or anything.
No, it's not going to really mess with that up. It's basically,
Okay.
something happening to the chemicals in the ink that it sort of
Comes
fades away or disappears. But look at all the old collections from herbaria anywhere. People wrote stuff on paper with pencil, and it's still there, and it's
Yeah.
readable, legible.
Cool.
But
Okay.
I would prefer just ink, and like I said, all my index cards from 8 years ago, 10 years ago, they're still in great shape.
All right. So
and
good.
I
keep
just
keep
use
a,
little
keep a
pieces of
spreadsheet.
scrap paper and
Yeah,
write
Dave
down.
does that.
Yeah. And but I usually try to include an MO or an INAT number.
Oh,
Oh,
absolutely.
perfect idea.
MO number, name of the mushroom, the proposed
Yeah,
ID, date
yeah,
collected, where it was collected, and who collected it.
yeah.
Right.
If you have the MO number or the INAT number, all of
Then
that
the other
information
stuff.
will be automatically available
Right.
to anyone with a computer. Um, Also, I'd like to suggest also when you package a specimen in a Ziploc bag, try to squeeze the air out of it.
Oh, okay. Good
Correct, yeah.
information. Yeah.
In my experience, storing mushrooms at room temperature, especially boletes, did not adversely affect them. They did not get moldy or wet. They were still very crisp. But other mushrooms, like some Amanitas, for instance, do pick up moisture, and that can be detrimental to the quality of the specimen, especially if you're going to get it sequenced.
Yeah, I am
So,
concerned, because all this, everything, like my little lab is in the basement, and I mean we've had so much rain and it's so humid. Even with a dry specimen, I'm just, yeah, nervous, but I kind of have to do it down there.
right,
I'll,
and this is
I'll
not
throw
something that I recommend, but I put whatever I could fit in my fridge. Those shoe boxes, I did that with probably older
Uh,
collections.
that's a
And
good idea.
yeah,
Yeah,
not only the refrigerator keeps it cool, refrigerators are also
right.
naturally dehydrating. So you will be
Yeah,
constantly removing moisture from that air. So that's going to keep stuff really, really in good shape for a very long time.
that's good
Not
information.
the freezer, refrigerator. Okay, that's the difference.
Yeah, yeah. No, I know. Yes. Okay, cool.
that,
Um,
Hunter, I'll throw one more thing that has really changed because I just have my little herbarium, fungarium in a whatever you call it, a tote, right? A big plastic container. Um,
that's also a good idea. Yes.
and I just bought, a thing of salt, and the entire bottom is salt.
Oh
In
boy. Uh-huh.
And that seemed to have made a difference because I was getting some specimens that I dehydrated and they were kept in my closet, not the best place. Uh, and they were getting a little gummy when I would come come
Yeah.
to check on them. But since I added a nice thick layer of salt to the bottom of these sealed bags so the salt's not getting in there, that seems to have helped a little bit.
Cool. And just even the tote alone, like the large tote with the shoe boxes inside, I mean, I think would be helpful. You know what I mean? Rather than just the cardboard boxes exposed
Yeah,
Yeah.
air.
Well,
yeah.
I
Yeah.
did forget to mention that I do have plastic boxes that I bought from Walmart a long time ago. They have actually rubber seals, and they have these
Mhm.
clasps. So the rubber seals are pressed against the rim of the box. So they
Yep.
are for the stuff that's supposed to be kept outside. So and then I threw in like a bunch of these silica gel packets inside. So I think
Perfect.
it's a fairly dry environment. Also, especially that when you squeeze out all the air from those bags, yeah, it really keeps things dry for a while.
But
Okay, cool.
important stuff goes in the fridge. Yeah,
The
the
important stuff goes
individual
in the
bags.
fridge. Yes.
Yeah.
Okay.
Yeah.
Okay, good. Thank you so much.
The
I
other thing that I think Jason was trying to say is keep a spreadsheet, have an index
uh
number so that you can find them, have some
Yeah, I
sort of
mean,
a catalog.
well, okay, thank you. Yeah, like I
you don't have to be 80 years old and be getting ready to submit your thing to a herbarium and they're asking for a spreadsheet. You got to go through everything and type everything in. It's a lot easier to do it as you collect it. So
And by the
I
way,
mean,
yeah.
I don't have to worry about keeping things sealed with lichens and bryophytes and stuff. So I have a spreadsheet that, I plug everything into the columns and then it automatically generates a label for me. And then I just
oh
print it and it folds in a way so that when I fold it, the label is on the flap that comes over. Um,
oh
but yeah, if keep a spreadsheet so that you don't have to enter everything all at once
okay, and then
years
that
down the road.
brings me to, so when you have these boxes, are they like, do you Do you put them by genus in alphabetical order, or?
Yeah, that's preferable. And, I would like to comment on that. And I would also like to add to Jason's comment about labels. You can actually create observation lists on Mushroom Observer, and you can pull everything, say, by year or by a specific place. And then you can recall these lists, and those are going to be duplicating your, say, Excel spreadsheets. And this is, probably more preferable. But you can also, as far as labels are concerned, there's a feature on MO. You can print a label for any observation that's going to include a QR code, but it's going to give you a basic information about the mushroom, the name, the MO number, the collection location, the collection date, and who collected it. So you can print them out and on that acid-free paper and then put them into your bags instead of doing everything by hand. So
Cool. Yeah, that's nice.
right. And what's the other thing that Jason? No, not you said. Everything in my herbarium has been stored by year. And I just a day or two ago, I had to go through a bunch of collections of mine from multiple years to fish out boletes that Colin Damnauer wanted to get sequenced again. Okay, this is like my 10 for 12 package to him in the last 4 years. So, and I it took me like half a day to find these things, and I said I should probably organize everything by genus. And whenever I need a Tylopilus, I'll just go to one box and it's going to be all full of Tylopilus.
Yeah, so
As
that's
opposed to looking for a Tylopilus collection in a box that's full of Amanitas, Rusovas, boletes and other stuff.
Yeah. Yeah, and I mean, I've kind of gone through this in my photo files as well. I used to do by year, and then it's like, what?
Right.
Yeah. So now, yeah, okay.
Yeah,
I have no recommendations for you there because my photos are a mess.
yeah. The problem that I have with organizing by taxon is that you got to start off with a lot of shoe boxes because it's difficult to organize everything.
Oh, right.
So I did
Wow.
an email at work for people. If anybody's buying shoes, I'm looking for shoe boxes.
Hysterical.
So I have a whole stack of shoe boxes now that I can start, just putting a family on each one. So I have a lot of shoe boxes, but a lot of them are, only have a handful of things in them.
That is so funny.
Well, you
That's
could
good.
just use alphabetical. These are my L boxes of taxons if you don't have a lot in one shoebox.
Right. You certainly could have multiple taxons in one box, but
Yeah.
right.
Problem is I've got mosses and liverworts and lichens and
Yeah.
we'll try to keep them all split.
All right. Well, we know where to bring our shoe boxes.
All right.
And I
So
don't want to take away from the add on to this conversation too much, but I do locations, and as long as you have an observation at that location, you can find whatever you're looking for. If you look up your own observations of that thing, you can find where it was, and then
Right.
go to that box. So.
Oh, cool. Yeah, that could be useful. Okay. Well, thank you so much, and I definitely want to start getting my herbarium in order. So I have just another observation or two. This is Aureoboletus annexus. I'm pretty positive. And because actually I had shown it from the same location, and that's what you guys said. And then I went back and found some more. Is that the consensus? And it's a group. It's a
Probably, but it would help to harvest with the entire stipe intact because this species has a distinctive tapering stipe that's pinched at the bottom.
Okay, it did have that fresh, what I think maybe
witch hazel odor.
Yeah, that witch hazel smell. Yeah. And I do have it. But yeah, and I did harvest it that way and then cut it off, of course. But yeah, I don't know why I don't have any in situ pictures right now. But okay, but probably probably. And
So yeah, once again, caution. Yeah, this is in the innixis group as we understand it in a modern sense. But even that we it's been shown that we have more than one entity that can qualify as innixis. So
right.
you have to be careful. But this is still work in progress. But this is as far as we can go.
Okay.
Call it innixis and call it today.
All right. Okay. And I think I'll just show this agaricus. I didn't rub it. I didn't do anything, but I just wanted to show one of these reddening agaricus. But I have no clue which one it is, and I did smell it. It was mild. It may have smelled like almond. It was not a bad smell at all. But it was pretty, and they're all over.
Can
So.
we zoom in to see the caps better? Is it really true reddening, or this is part of the overall color scheme of the cap? These um, how do you call them? Not scabers. Geez.
Areolate or no?
Yeah, well, I mean these pigmentations, on the cap, these brown pieces, not warts, whatever you call them. I can't remember the word, but is it?
Fibrils? No.
No. Fibrils are more like hairs. These
Yeah.
are like more like patches.
That's areolate, isn't it?
Yeah, but so this pink, is it really staining, or this is just a natural cap color? That's two different things.
Well, it starts white. I mean, as a baby, and then they become this. So I was almost wondering if it's like a reaction to heavy rain and H2O, because it seems like they start white, and then as they get older, then they become this pink.
Yeah, I guess you can get a staining reaction from the elements. The water pelts the caps, they get soggy, and then something happens, and you bring out the color that you would normally get by disturbing the mushroom by breaking off a piece or by scratching it. So this is certainly believable that this species actually is a red staining, brightly red staining agaricus. Um, for a moment, I thought maybe this is Binosobrunnifumidus, just because the way the caps are ornamented on the disc, where they're brown, breaking up into these scales, with areolations, and the fact that this is growing under conifers. This is where you would mostly find Binosobrunnifumidus in the pine barrens like habitat, where you have sandy soil and mostly pines around, with some oaks maybe here and
Okay,
there.
so this is true. It's under a few white pines, but it is predominantly oak, and you can see oak. I think that's an oak leaf in
That's
the
an oak leaf, yeah.
right. Yeah.
I don't think this is Binosobrunnifumidus on the account of this bright red staining. That species doesn't stain red. But
Okay.
other than that, it kind of looks similar. But you can say the same thing probably about a bunch of other species of agaricus. It's a very difficult genus.
Right. Okay.
How big are these things?
Good size, probably almost 4 inches across the cap,
Hmm.
and then maybe 5 inches tall, 6 inches tall.
Yeah, they're pretty.
Yeah.
I have absolutely no doubt that if you presented these pictures to Rick Kerrigan, he would tell you conclusively what the species is. Also, look at the base. It
Yeah,
seems like
I was
the base, the
thinking.
bulb is marginate.
Yeah,
Marginate.
I was thinking someone has to go shake him up and pull them out and just say, no one's going to buy, we're not going to all buy that book. You have to come back.
Yeah, the last time, I think we heard from him, he said that pretty much verbatim, quoting him that he wrote or he taught everything about agaricus that he wanted, and there's nothing for him to do anymore in that field. And he switched completely to some environmental science group
Right.
or whatever. He is doing research in that area. And if you try to bother him with agaricus, he's probably going to snap at you or something. Probably not.
But these are the ones that he would have liked you to collect and get sampled, because the red staining ones, they don't have enough data.
Oh, okay. All right, good. Thanks, Sue. And maybe I'll try to get some more information on them. Thank you. Thanks, everyone.
Thank you, Hunter.
My pleasure.
Hey, Shihong, you're the first of the emails.
Okay.
Let me get my two.
Yeah, this is the weird bolete we found at the last foray.
Ay.
That says Noel, I think.
Yeah, I think I have more picture. You can probably see the next one.
From Shark River, huh?
Yeah.
Looks like it's hypomicized, and if it's hypomicized and it looks like this, it possibly could be Xanthogonium athene or purpureum. Maybe.
I feel like I handled this thing and it didn't seem hypomicized. Let's go to the next.
Yeah,
let's go to the next
see the
photo.
next few photos. The last one, maybe. The last one has 4 or 5 of them.
Roll here.
Oh yeah, definitely hypomicized.
Oh yeah. Yep.
Okay. Yeah, yes. Okay, so our guess is Xanthogonium athene.
Well, let's go to the last photo. Let's have a look at the last photo.
Yeah, let's
you want to see more?
Photo and expand that. Yeah. I have to rotate my computer to see better.
Yeah, rotate your head.
These are Xanthogonium athenes. Yeah, these are Xanthogonium athenes. Yeah, you can see in the larger fruiting body that's up front that it still has the looks of Xanthogonium athene. Yeah, they become this, club-shaped, disfigured entities like you see them here, and they smell awfully bad, like dead fish.
I did not smell anything, actually.
Lucky you. Because once you smell them, you cannot undo that experience.
Okay, so it's Xanthogonium athene.
Athene, or you can basically put the name of the hypomyces, which is probably Chrysospermus or Megalosporus. I forget. I think they're very host specific sometimes. So yeah, Xanthogonium athene.
Okay. Oh, this is the photo did not take the color. The color in situ, it's kind of green, and it's very slimy.
Looks like one of these Hygrocebi genera.
Gliophorus, maybe?
Gliophorus, yeah. There you go. I thought it was a mycena, but once you said a green color, I said, no, it cannot be a mycena like that.
So what's the name? Can you type in? Is that
Gliophorus genus?
Yeah.
Yeah, let's see. Is it that with a Y? G-L-Y?
G-L-O-I, I believe.
G-L...
G-L-I-O. I'm sorry. I think it's I.
It's an I, not a Y? Okay.
Nah, it's an I. So if it was green, but the gills should also be a little green or yellow for Piscitaceus, however you say that. The parrot. wax cap.
Pseudosinus. Yeah.
Okay, there you go. All right.
Well,
The
there's
parrot
also
one.
there's also that perplexus, right?
Perplexus. Yeah, that one has weird colors too. Yeah. And those are so slimy, they're hard to pick up.
Yeah, it's very hard to pick up. And
On the stipe and the cap, right?
Yeah.
Both are slimy
yeah,
because I think that's the distinction there.
Yeah.
Galeophorus.
Just moving on quickly because I know we're getting
Yeah.
late.
And you found that at the foray at Shark River, the Gliophorus?
Yeah, and I kind of did not even took out my basket. When I dumped my basket, it's stuck between the
Yeah, that's that's funny because
Yeah.
when they get to the table, they're almost indistinguishable as
Yeah.
Gliophorus because they dry out and things like that. So
No, they did not dry out. They just stuck between the branches in my basket, so I never
the
even took
wicker,
it out.
yeah,
Yeah.
the wicker,
Yeah.
yeah.
Yeah, here's our friend that we mistakenly called Bolletus Olivosporus, and then because of genetics, we came up with a new combination, Rufoboletus Olivosporus, except that this is not Olivosporus.
Rufoboletus.
As I showed last time, the description of Cereomyces / Boletus Olivosporus by Merrill, the original description, indicates that it's a very different species.
So I'm going to send the Burkharts an email later tonight. What do I call this thing? Boletus Olivosporus?
No, I mean, call it just Boletus Olivosporus. So we know we're talking about the same thing, even though it's a mistake. It's a misnomer
Yeah.
in many ways, in more than one way. Just give them that name. We'll deal with that problem later when things get published.
Oh, what's the species name again?
Well, like I said, phylogenetically, this particular bolete, the species, falls into a genus called Rufoboletus.
Oh, okay.
Yeah, RUFO,
Okay.
Rufoboletus.
Okay.
And the new name that we would like to propose for this species, because we want to believe it's undescribed, although it's hard to believe it's been undescribed, because it's such a common bolete on the coastal plain from New Jersey down to Texas. The name that I want to give it is Rufoboletus mutabilis. But you don't have to really remember that name because it's not published yet, and this is just all kind of a little bit of a pipe dream at this time.
Okay.
But yeah, what you saw in Kuos, yeah, I'm not sure where that mistake originated, and I believe it was the Besets who promulgated this name and slapped it onto this bolete for reasons that are still unknown to me. Because
So does this species bruise blue on the pores and then it changes brown?
it's on the flesh. This bolete stains blue, and then immediately within basically a minute or two begins to change to reddish brown, and hence the name for the genus Rufoboletus.
Oh, because I noticed on the pore surface I saw some blue and some brown.
Yeah, I mean, this is really sensitive. It stains, it bruises blue. I mean, you could basically drag a hair or a grass blade across the pores and I'm going to stain blue. That's how sensitive the hymenophore is. And when you cut it, it's going to be marbled like that, and it's going to stain blue, but not very darkly like, say, Miniatopalescens or like Neoboletus or Cyanoboletus, but it's going to stain blue like this on the context, kind of a medium blue, but it's going to be uniform, and that blue is going to be disappearing and replaced by reddish brown color like the ones you see on the pores. So a very interesting feature, very distinct bolete. It's beautiful when it's young and fresh, And when it's older and handled and weathered, it turns into this uninteresting, cryptic, all brown, drab colored thing. And everybody's like, what is this? So hence the name that we want to propose, Mutabilis.
Okay, I think next one should be last one.
Let's go for these pictures again, please.
First one.
Yeah, let's zoom in on the very interesting.
Was that was Shark River, right?
Yeah.
Very interesting.
Is that one of those longicurvipes, but it's all beat up?
But the pore is kind of big.
No, this is not Longicurvipes. This is not Leccinum.
Maybe Porphyrolis.
I thought maybe this is Sordidus, but the stipe is too yellow.
Yeah, the stipe is light. Yeah,
And the
Porphyrolis
cap
has
is
dark
too
stipe.
the cap is too orange.
Yeah, that too.
Can we go to the next picture because that's the one that shows the pore surface better? Yeah, let's zoom in here. You see, it has a xerocomoid pore surface. You see how thick those tube walls are, and how fused they are with each other. And I guarantee you, they're not gonna break apart cleanly from each other. They're gonna snap. Each tube is going to get mangled when you pull them apart. They're gonna disintegrate into halves, not whole tubes. I don't know what this is. I wish I was there. I wish this specimen was saved. But
I don't know if it was.
It's not Tylopilus. If you think this is like, okay, the pores are kind of whitish and then they stay in this, brown. The stipe is too yellow.
Could
And
it
Tylopilus
be
has boletoid.
xerocomas?
It's not a xerocomus. Xerocomus would not stay in brown like that.
Did this stain immediately brown, or is this later and there was a blue
I think
stage
it's
before?
later. I forgot what it looked like.
So this was not saved. It was tossed away.
Probably because the last few things this is on the table. I cannot ID.
Yeah, this is very interesting. I don't know what this is. I think this is the first time I see something like this. Albeit, admittedly, maybe some of the pictures are sort of distorted in their colors because of the light and the way smartphones interpret those kind of lighting conditions.
So the orange cap, definitely it's true.
Yeah, the cap is orange. I see that, and the stipe is yellow.
And it's super buggy on the third photo.
Yeah.
Let's see the cross section again. Maybe we can get something from that too.
That flesh white or pale yellow?
Kind of white.
Yeah, it's off white, and the bug damage turns the flesh pink. Yeah, I would like to agree with Dave that maybe this is something in the Porphyrolis Tylopilus, but I'm not convinced. It's just there are too many things that contradict those generic assignments.
Yeah, well, I'd like to agree with Dave too, but for the same reasons you're giving. This
you would like to you're referring to yourself in the third person now.
Okay, Xiang, you have one more, which is
do I have one more?
Yeah, you have it.
I'm sorry. Okay, this one. I saw their Gyroporus, but they are huge, one of them. So I thought Gyroporus should always be small.
It can be big. It can be big.
Yeah, there's this Gyroporus.
Oh, but look at the stipe. Look at the inside of the stipe, though.
Yeah, no compartments.
what is it? Rosy? Oh, that looks like it has
This is Rosea
scabrous.
albus, I think. That's what this
Yeah, I
is.
was going to say, do you think Roseo albus
Yes.
is pitted?
You can
Has
see
the
that the flesh
cham-
begins to crumble away, so it's like it's not chambered, but it's pithy enough or it's crumbly enough to indicate that it's going to become chambered when this thing gets old.
Okay, so
Um,
that's the pithy stuffing inside the stipe.
right. This is kind
Yeah,
of
what you want to do is see if you can snap the stipe shock. Gyroporus species have the cells arranged in circular fashion around the stipe, like latitude lines. So they snap apart.
Well, this does look like it was snapped and not cut off the cap.
It does. It does look like the stipes were snapped right off and snapped off pretty cleanly.
I would like to point out that for a long time the species has been called Saproamanita, and that's a real name. Both of them are real names, and both of them are Murrellian names. They're by William Alfonso Murrell, who was very active in Florida since the late 20s, early 30s, up until his death in 1957. So he found a lot of boletes, and he described a lot of boletes from Florida, and we don't know much about them because they've been very resistant to sequencing. Unfortunately, the conditions in Florida, all that humidity over the many decades of storage, have pretty much destroyed a lot of his collections. They are sterile. It's very difficult to get DNA from them, and that presents a problem. So, like I said, for a long time, it's been called Saproamanita, but I think one of the more recent dropper studies by the last student of Roy Holling, Navid Davudian, they had good inferences from their analysis of type specimens that the proper name of that pine barren species that we find so often in New Jersey is most likely Rosea albus rather than Saproamanita. But I welcome you to read the original descriptions and see how much information you can infer from those, because that's the point of origin, and that's always very important when you begin chasing taxonomy and nomenclature in mushrooms.
All right.
Okay, so do we have a species you want to suggest on this one? Does it have?
I would put Roseo. I think it's Roseo albus or it's Rosea albus. Start typing. Let me search that in Index Fungorum to give you the proper spelling.
Okay.
It's not Roseo albus.
So can you
Or maybe I'm just mistyping.
Yeah, I'm just going to do that.
try to put it in down there, or just go propose?
Oh, sorry, I am logged on, but
maybe it is Roseo albus.
Gyroporus R though, right? Rosie albus.
Roseo albus, and I'm not
This is just
finding
an
out.
ROSEIA.
It is ROSEIALBUS. Rosea.
I put it in already.
You put it in?
Yeah, I put it in.
Roll down.
Okay. All right. Terrific.
Thank you all.
Okay. Thank you, Shihan. Okay, Dave. You're the last.
Yeah, Jason, are you still here?
Yeah, I'm here.
Okay, you know what?
The
Start
lichen?
with that very last thing, the lichen.
Okay.
So it's on a little piece of bark. It's hard to say much about it in this sort of big picture. But I've got some photos through a loop that I edited and tried to sharpen up a little bit. And so we see the apothecia on these subsequent photos. You can't really tell much from this photo, I don't think.
Yeah, I'm trying to get back to the observation again.
Oh yeah, get back. Yeah, so
Okay.
these photos were taken through a loop. They weren't the greatest, and I tried to edit them and sharpen them up a little bit, but you can see the apothecia. It's kind of this light brown color. So I guess I think I proposed. I mean, Karen didn't know what it was, but I think I may have proposed Leccinum as just a low confidence.
Yeah, so you can see, so keep it on this photo. So you know there are three main types of apothecia. Well, one of them is called a Leccinoran apothecia, which is named after the genus Leccinora. And those apothecia have rims around the margin of the apothecia that are different colored than the center of the disc, but the same as the color of the thallus.
Okay, you know, she told me she looked for that and didn't find it.
So that suggests this probably isn't a Leccinora.
Okay, so Leccinora is probably wrong.
The second type of apothecia is called Lecidine, which is named after the genus Lucidea. And that is usually, it looks like a Leccinoran apothecia. It's got a flat top. It's got that rim around the margin, but the rim is usually the same color, very similarly colored as the center, which this doesn't look like either. So I think this is the third category, which is a biaturine apothecia, which is basically rimless, and it's usually not flat on the top. These are just kind of like these, they look kind of inflated.
Yes, they do. And there's no rim. Right.
Yeah. So a biaturine apothecia usually points to something that's either in the genus Biatora or Basidia, like Basidia schweinitzii, the champion crust of Eastern North America. But usually Basidia have darker colored apothecia. So I think this is probably in the genus Biatora.
Biatora.
Biatora. And if I had to guess a species, I'd say Longispora. So I could put that in the chat.
Oh, you know what? We looked for spores. I didn't get any photos. It looked like the spores were pretty long, if that's what actually was found. And they were kind of, I didn't get a photo. I should have tried to get a photo. And I think they were sort of like elongate. What's the word I want? Fusoid, subfusoid.
Yeah, so Longispora is a good guess. It's a common species, at least here in New Jersey. Vernalis can look kind of like this, but usually Vernalis, I think, is on moss, not on bark. So you usually find it terrestrially. So I would start with Biatora Longispora.
Okay, thanks. Well, we got something to go on now. I think she was kind of stuck on this one.
Well, I got to the point in the key where it said, is it P plus or P minus?
Oh, yeah, she got to the point where it said P plus or P minus. So what does that mean?
P is the chemical P, which I don't have.
Oh, that's that one, that carcinogenic one that you don't want to get. Okay.
It's on a list, maybe.
So what's a P?
Excuse me?
What's that P? What's the name of the chemical?
What? She doesn't even know.
It's p-phenylenediamine.
Oh, it's an aniline. It's an aromatic thing. Yeah, those are nasty.
Yeah.
So I accompanied Hannah Huber on a survey of a old growth plot in the Pennsylvania State Forest that she's trying to get a species list for, and she found some of these and she took them to get them sequenced. But then there were a few more on some fairly well-rotted wood. So I took a few because I thought I knew what they were. I didn't remember the name, but I remembered there was like TAR in there someplace. Or TAP, I forget. But anyway, but the name is posted on here. Now I'm pretty sure I got it right. Based on what these look like, they're small, these small cups growing on well-rotted wood. And the spores really kind of give it away. They have really big spores. Some of these spores are, I think I found one spore that was like over 35 microns long. It might be this one in the upper right here. There might be there's probably another picture of spores. Yeah, okay, here we are, the really big ones, the one in the lower right. It's over 35 microns long. That's pretty long. So I'm pretty sure I got this right. I found it before. And I think the actual species name is like Megalospora or something like that.
Macrospora.
What is it?
Macrospora.
Macrospora, right. Yeah, macrospora. Yeah, because the spores are so big. So these little guys always have the biggest spores. So that's these on well-rotted wood.
Dave, that's not the old Alvella macropus?
No.
Okay. Different thing.
Different thing.
Cool.
Yeah, Helvella macropus is larger. It has a longer stipe, and the spores are going to be a lot different. Helvella spores they have a much smaller Q value. They're not as large. They go up over 20 microns. Usually they have one great big gutule. Mm-hmm. So it looks like this giant eyeball kind of.
Okay.
They're grayer.
Yeah.
And I don't think they grow in the same fashion either.
The Helvella, some of the Helvella grow on rotted wood, but usually they're on the ground. You're right. You're right about that. Usually they're on the ground. They don't have spores that look like this. Spores are really a lot different.
Oh, yeah.
So this is a dead ringer for Cuphophilus canescens, except they are so tiny. Look at these guys. They're so small. Otherwise, they look just like Cuphophilus canescens. So I put them on INAT as Cuphophilus canescens. I sampled for DNA. So I'm pretty sure I'm getting a lot of results from my submissions to my CODA lab using that toothpick technique with the wells. But this looks just like Cuphophilus canescens, which is usually, a fairly good sized mushroom. Maybe because it had been dry for a few days, these just didn't get very big. I don't know. So I thought that was kind of interesting. In sort of dry moss, in more or less acidic environment on top of a mountain. There's it's really sort of like a plateau up a little bit northwest of where we are here. But it's a very acidic environment. So those are pretty cool, I thought. Well, so I'm not sure what these are, but I think Cystolepiota makes sense. They're growing on litter. There's apparently a saprobe. Not a well-defined ring, but there's a lot of deposits on the stipe. I think one or two of them may have shown a faint ring zone. Spores are white. Spores were not dextrinoid, so I mounted spores in melts. I put melters on the spores. And if they were dextrinoid, it was very, very weak reaction. So a lot of the lepiotoid fungi, Lepiota, Macrolepiota, Leucoagaricus, Chlorophyllum, I think a lot of those have dextrinoid spores. So these are mounted in melters, and they really have not. They didn't darken very much, and they're very small spores, and that fits Cystolepiota. Very small, thin spores. So I think that's what I have here is Cystolepiota. So I sampled them for sequencing, but it's something that I haven't found very often. So I thought it was kind of interesting. In mixed woods,
Oh,
going on forest litter.
I'm seeing a lot of other little lepiotas too.
Yeah, I found some. I think I have a lepiota in here someplace in one of my links, because I found some lepiotas also. So yeah, I thought those were kind of interesting. So also in this tract of state forest where we surveyed, Hannah took this. She'll dry it out and send it to my Coda Labs. But I thought this was an interesting little section vaginate Amanita. I mean, it's a beautiful specimen. Look at how nice the vulva has come apart. There's no deposits on the cap.
Lovely.
It's all in the bottom. Yeah, I like this because it looks so nice.
It's,
The
And
baby
it's
in the
not.
bonnet.
I don't think it's vulva. What's that?
Looks like a baby in a bonnet.
Yeah, yeah.
Sweet. It really is. Is it as small as it looks?
So
Yeah.
it was. Oh yeah, it's small. It's small. Look at there's centimeters on top there on the field voucher slip, so you can see that. I mean the cap is. If the cap was stretched out all the way, it might be about 2 centimeters. If it was expanded, so it's a small mushroom. Now it might grow. It might just be it's very because apparently it had just recently burst through the partial or the universal veil. With the nice striations on the cap. It's not fulva. I don't think it's Sinica flava. It's the wrong color. It's not. I don't see any gray on the vulva. Oh, a really young one like this, the vulva might not be graying yet. I don't think it's one of the stirp sorocula things. I think it's just some sort of section vaginate, series vaginate, whatever it is that Rod calls them, just a regular sort of grisette. But very nice mushroom. I sampled it. Oh no, I didn't sample it. I'm sorry. I just photographed it. Hannah took it. She's going to dry it out and send it. So I hope we get some feedback on that one. Oh, so here's the Lepiota. So I think this is Lepiota felina.
Yes, I would agree.
Yeah, very dark scales in forest litter. Nice persistent ring on the stalk. I didn't even look at spores here. I guess I just had too much to do. I get sort of overwhelmed with how many mushrooms I bring home, and I fall behind, and I don't have enough time to do everything. But I'm pretty sure this is Lepiota felina. I sampled it.
So
I'm
what
sampling almost everything I bring home, except for stuff that's like totally boring and easy. And sometimes I make a mistake and bring home something that I think is boring, and then I find out a little later, oh, there's like 5 of those. I should have sampled it. But anyway, I did sample this.
Right. That's it. 2, 3, 4, 5, right? And that was just.
There might be another one. I don't know. I forget.
No, that was
what I
it. That's
showed.
the
Oh, that
5th.
was it. Okay. All right. Thanks.
Thank you, Dave. Any other?
By the way, some of these small lepiotas are thought to be dangerously poisonous.
Okay. Anything else to share before we close for the evening? Okay. Well, hopefully we'll have plenty next week because we're getting more and more rain this week.
Yay.
I have a couple things to share if you want to keep the
Yes.
meeting running.
Going. Yes. Be happy to.
If anyone,
Are you mailing them in or?
no, I can share.
Okay.
Can you guys see my screen?
Yes.
All right, and then I was just on a Bolletus Oliviosporus, and I put it in the chat. I think I put it on the ID as pretty confident. So, Igor, would you agree there?
Oh yeah, it's a misnomer, but that's what we call this thing at this point in time.
Yeah.
And you can see the fresh cut that's blue and the old cut that's reddish brown.
Right.
Boletus.
All right, cool. And that's why I wanted to share, just to kind of confirm or get some information on some stuff that, you weren't there. So we had a bunch of Amanitas and Boletes that I'm trying to ID, which is not great. I'm calling this one Xericomis illudens. I think this is probably the best photo of the kind of reticulation, elongated reticulation on the site.
Yeah,
Probably something
more
in
like
that.
ribs, really.
It looks actually elongated reticulation, fairly coarse. I think this is something from the 10x group. I don't think this
Okay.
is illudens. Illudens is more graceful, and the cap
Okay.
color is different. So, and it's
So,
not as reticulate.
so maybe a correction on when I send this email to the Bark Arts, maybe 10x group
Yeah,
instead of
10x.
illudens.
Yeah. Mm-hmm.
Okay, cool. We'll just look at this one real quick. It's not a good observation, but this is the other specimen of Aureoboletus innixis, or at least that's the name that I used. I guess group, but
Yeah, this looks more like Aureoporus to me.
Yeah, I was going
Okay.
to say Aureoporus. The stipe is wrong for Innixis.
Yeah, the step is wrong for Nixus. It's kind of translucent, like ivory kind of thing, and it's flushed with red, not brown. Yeah, this is something in the Aureoporus group, which is also a group species. A complex of species, because there's more than one, and we don't know which one is the true Aureoporus.
Okay.
But yeah, this is not a Nixus.
Yeah, I would call it Aureoporus.
Yeah. Mm-hmm.
Yeah,
For the
Innixis
time
has that bulge in the lower, like 2/3 of the way down the stalk. It usually is fairly well bulging. This one's a little thicker, and it doesn't have the pinched base on the
Doesn't have a
stipe.
pitch base, and the cap colors are wrong. This one is more
Yeah.
reddish than brown, and it's more streaky. And if you look up observations of Aureoporus or pseudo Aureoporus from the southeast and the Bible Belt, you'll see what I'm talking about. They have this unmistakable look about them.
All right,
So
cool.
yeah, how big was this? Not very big, I presume, right?
It was the exact same. I'm telling you, it was the exact same size as the Innixis. So
Okay.
probably, 4 or 5 inches.
Okay. Yeah, Nina collected something that looked very similar to this at Wells Mills County Park a couple of years ago, and I preserved it. And that was also an unusual Aureoporus type thing, but it was quite big and chunky, and not quite the Aureoporus that we collect. That's usually smaller and has a viscid stipe and viscid cap. Mostly viscid cap and also viscid stipe. Not so much, but it has the same quality to it. The ones that Nina found were completely dry, but they were in that group as well. And I don't know what they are, but they're certainly not in Nixus, and they're closer to Aureoporus. And yeah, this is where we need to do more sequencing, and maybe we'll find something additional, additional taxa in Aureoboletus from the Aureoporus group. Very nice. Like I said, I wish I was there to preserve some of these more interesting collections like this one. And also Shihong's unusual bolete with an orangish cap and whitish xericomoid pores that stained brown. reddish brown, maybe even yellow stipe. That was very interesting too. Cool.
Yeah. And then so this section vaginata, it is grown in moss, mostly hardwoods around. It does have that kind of grayish coloration on the UV and the PV.
Pachypondensis.
That's what I was thinking, Pachypondensis or maybe Rhacopus, but
No, this is not Rhacopus.
Yeah,
Okay.
not Rhacopus.
No.
Yeah, I don't know what this one is, and I don't know Pachypondensis.
Yeah,
so.
Pachypondensis is battleship gray color of the cap and lighter gray warts. But you can see this is from the stirps or series Sororcula or series Siciliae, I should say. That's a more encompassing taxon in terms of number of species and what they look like. But yeah, all the ones with the gray or graying universal veil fall into series Siciliae. Stirp Sororcula is more precise, centering around Rhacopus, I think. Yeah, I would say Pachypondensis. Very good chance.
Yeah, so Rod had a name for Rhacopus before the Canadians named it Rhacopus. He called it Sorocula was in the name. There were a few more syllables. It was a really long name. It meant like little Sororia sister or something.
Sister, yeah. Mm-hmm.
Yeah. But that's where the Stirps Sorocula name comes from.
All right, cool.
Yeah,
So
these are called strangulated amanitas sometimes because the vulva, the universal veil kind of disintegrates, but it collapses. The top part of it collapses on the stipe base and strangulates the bottom of the stipe.
Yeah, Amanita varicolor has that kind of a vulva. Actually, it leaves a ring on the bottom of the stipe that's flaring. It's like a little bit of maybe of a funnel shape, but it has that feature, and that's an example of strangulated vulva. It doesn't have a true sac. The rest just disintegrates, but it leaves this sort of annular-like structure, like you see here on the bottom of the stipe. Kind of a rough-looking ring, but it has sort of an annular, appearance to it. A little bit more organized than something which is just like a bunch of oppressed pieces of universal veil, friable pieces of veil clumped together. But here it looks more like something that's more organized and some kind of a annular feature. And the limbus internus of the vulva you can see on the stipe as these bands, gray bands, zebroid or chevron-like. That's the internal part of the vulva, the internal layer. Yeah. The more I look at this thing, the more I'm thinking this is Pachypondensis. It's quite small. It's not a very large species.
Little dainty little thing.
Mm-hmm.
Cool. Yeah, I didn't want to use that name because I don't know. Seems like a not often used name, but probably not very uncommon around South Jersey and these kind of sandy soil mixed woods.
It is from that habitat, and I don't think it's found outside of it, to my knowledge.
Yeah, I was going to ask if this is found anywhere outside of South Jersey.
Or we should say outer coastal plain.
Yeah. Isn't there a supported DNA match from Florida? I think there is.
From who?
From Florida.
Let me take a quick look. Byron is there. Okay, Pachypondensis. I'm just checking observations here quickly. Here we go. Okay. I think all of the observations of Pachym pandensis on Mushroom Observer have come from New Jersey.
Wow.
6 of them.
And you'll be happy to hear I did save this one. So if we, it is
Yeah.
dehydrated. So if we want to do
Excellent.
something, yeah, if we want to do something with this, we can.
That is really interesting.
So I'm at Rod's website. Just want to see if he states, collections. Yeah, they're all from New Jersey. Although similar to A. racopus in macromorphology, the present entity appears to be genetically distinct. The smaller spores, the relatively large velar patches on the cap, and flaring limbus internus. Remember I told you about the flaring ring, the funnel shape thing?
Oh, yeah.
That's what he
Yep.
refers to.
Right there.
And or the upper part of the outer limb of the universal veil may also be distinguishing characters. So there are good photographs. I'm going to actually leave a link here in the chat, and you can go to that page of Pachym pandensis on Rod's website, and there's a photograph of that species that looks exactly like Brandon's Amanita. With that
Beautiful.
flaring structure that's gray, sitting right above the lower portion of the base of the stipe. And once
Yeah,
again, because this is section vaginata, these will have bulbs.
wait, this is section. Vaginata. Yeah. I thought the ecology was interesting, just growing in the moss, but I guess it doesn't have anything to do with the moss, probably.
No, that's where the moisture is. My last Pachym pandensis came from Brandon Burn, and it was growing on the side of the road, like an inch away from the asphalt road in sand. So it just happened to grow there because it was wet, and it grew from pure sand.
All right, cool. So Aureoboletus group for this one. What was the determination on this? I didn't write it down.
Tenax.
Tenax. Thank you.
Tenacious. It has a very hard base of the stipe, and when it grows in grass, you really have to pull it out. It really grabs onto the substrate, hence the name Tenax. It's tenacious.
Tenacious. Beautiful.
There's 101. We have Tenax Indiana 01, which is in New Jersey, and that's what it could be. I haven't sequenced the real Tenax yet. It may be restricted to the upper northern tier of the United States. Huh.
Thank you. Yeah, so I'm calling this Hydropus. And it's probably Predicurrens. Not sure. Dave, if you're still here, I think maybe you know this one a little bit.
Well, I think I know it a little
obviously,
bit, but I mean, this seems to qualify for Hydropus predicurrens. Is it widely striate on the cap, like these wide sort of widely spaced grooves?
Yes.
Yeah. Okay. Yeah. Usually the what I did as Hydropus has really a lot of space between the grooves, but those are pretty widely spaced, and it is grooved.
And then,
that seems reasonable to me. This is the kind of thing you have to get sequenced, really. I think.
Right.
These little things. Yeah, but that makes sense. Oh,
and then,
you got spores. I forget what the spores are supposed to look like from this, but
Well, the Myco Quebec description describes them as distinctly amyloid.
Yeah, I was going to say I think they're amyloid, like a mycena.
Mm-hmm. But these seem more blue than black, and these are the spores, and they have this weird ornamentation without melters.
Oh, well, that I don't seem to remember. It looks like they have a gutule.
But
So,
then in
but
melters,
melters, amyloid reaction can be black or blue.
that's what I wanted to ask. As far as I know, amyloid can be blue, and these seem a little blue, but they're definitely darker than in water, which are these.
Yeah, I can usually tell amyloid reaction best by just getting a substantial spore print, and I put the melters right onto it, and usually the perimeter part of the melters, so it's like a drop that is like an amoeba, it spreads out, and along the boundary is where you'll see the darkest reaction for an amyloid reaction.
And I agree with you there, but the problem with this is, it's very small.
Yeah, it's hard to get enough spores.
So to get enough pile of spores, that spore print, these spores are from a spore drop right here.
Yeah.
And there's just not a collection of them enough.
No, you're right. It's hard to get enough spores.
But when I scope some material, I scope the material in melters, so you can kind of see the upper left quadrant there. That's just the material kind of taking on the melters, and the spores have a different reaction, right? They're not just going red.
Oh, yes. Right. Mm-hmm.
So I'm pretty sure it's Hydropus praedicurrens.
Yeah, it makes sense. That makes sense. I wouldn't disagree with that. Probably right.
Yeah, this shot in particular, that
yeah, and the gills are supposed to come down the stalk a little bit, like on those. Yeah.
Cool. I don't think we need clarification. Well, it's just us here, so let's look at the funnest fungus to find.
Oh, this is that thing that grows on dead mushrooms, right?
Yep.
Astrospora or whatever.
Astrospora, yeah.
Yep.
The russula in particular, blackened russula.
There we are. Yeah.
Astrospora Lycopredoides.
Yeah, Lycopredoides, right.
Yes, that's what you identified for at this foray.
Yeah, it's such a cool, weird, really weird mushroom, right? It's capitate, stipitate, but the spores form on the top of that thing.
Yeah.
And the pseudohymenophore is actually sterile.
Right. And when they actually open up, they have pseudo gills.
Right.
Mm-hmm. So the underside kind of looks like gills, but that's the infertile surface, the sterile surface. So just a weird.
These are pretty weird.
It is weird. Not only it's a parasite, it has gills that have absolutely apparently no function because it's not a spore-bearing structure, and instead spores form like a powder on the top of the cap. I mean, how weird can you be more than that?
Well, combine that with it's growing on a russula or
exactly. I mean, it's a parasite specific to a russula, but it's completely like backwards. I mean, this thing is like so weird. It's like, how did this happen?
Yeah.
What evolutionary forces shaped up this mushroom to be like that?
Yeah. I agree with all that, and I was trying to preach that at the foray, and some people were receptive to it. Others were like, "Oh, that's just a blob of old mushroom." But I didn't even do the microscopy on this because I'm pretty sure. I mean, that's what it is. But the microscopy is even more interesting with those stellate spores and all that.
Yeah,
Yeah, I
think
think I
actually
probably did it a long time ago. I forget.
it's one of the most fascinating mushrooms, even though it's not photogenic and very attractive. Just so many oddities and aberrations about it. Just make you wonder, like why?
Why?
Why
How?
does this thing
How?
exist?
How did we get here?
Right. What kind of a failed experiment was this? What kind of runoff, mycological experiment was this to create this monster? Fascinating. Do you know what family or what its nearest
Oh
relatives
God.
are?
Wow.
Have a look. Lyophilacea. How? How?
How
So that's
exactly?
that's Lyophilum, right?
Yeah.
Yeah. Oh
Yeah,
boy.
that's crazy.
My thought that this thing would be sitting on its own branch. It would be an orphan genus. It would be nothing closely related. But we've seen, real like weird things where Bunderzevia berkeleyi is actually in the Russulaceae or Russulalis.
Yeah,
Um,
it has spores with amyloid warts.
yeah, and phylogenetically, it's been proven that it is closest relatives are, Lactarius and Russula, as
Yeah.
opposed to Polypores.
Well, here's something in the same family. Sphagnoris palustra would be in that
Yeah.
family.
Uh, yeah. What family is that in? I don't even know. I know the
Lyophilacea?
species.
Yeah, the same family.
That's
It,
in.
it,
Oh, that's in Lyoph. Oh, really?
it,
Yeah.
it really sounds
Lyophyllaceous.
like
Wow.
this family is like a dustbin of,
Yeah, right. Everything's just getting swept in there.
Well,
yeah.
the Astrospora, I think that's a parasite
Hmm.
on or is it just a saprobe on dead
No, it's a
mushrooms?
parasite.
It's a parasite. The Sphagnoris paluster, that's a parasite on sphagnum.
Oh, that's a good
It
call.
kills
Yeah,
the
yeah,
sphagnum.
sure is. Okay. I can go down this rabbit hole on my
Yes,
own.
but your phone is going to die on you.
Oh no.
No, you got 15% left.
Oh, that's good for an hour too.
Oh, okay.
But that is all I wanted. I
Um,
don't think I have
wasn't
anything
there
else.
wasn't there, guys, another species of Astropora besides Lycoperdoides? I thought it
Yeah, I
was
think there's
fungal.
one other one. I
Yeah,
forget. I haven't found them in a long time.
yeah. I've seen only this thing once or twice, and my first time was at the Meadowood foray a number of years ago. I think I did not even know of its existence. I was unaware that such thing exists. Uh, or maybe I've seen this on the mouse somewhere, and I said, "Oh, finally, I find this. I see this in the flesh with my own eyes."
The other species is Parasitica.
I think that's the same species.
Quote keeps it separate.
Hmm.
That's saliva here.
Yeah, so what the heck is this thing with this tiny, tiny margin?
Looks
Or
like it's
context, rather.
Leccinum albellum.
Yeah, I agree.
Is that what
Just a
happened?
really old, blown out Leccinum albellum.
Okay.
Yeah, I mean they have humongous, pores sometimes. I mean, sorry, tubes.
Tubes.
The tubes are really long. Yep.
Yeah, but
No staining.
I sort of accepted the fact that this highly variable species is one entity, but I think we don't really have enough genetic um, evidence, and by that I mean multiple genes or maybe even whole genome
More than ITS.
to prove that this is one thing, but um, it's really like a chameleon in vaccinum. I mean, this
Yep.
thing can look many ways. It can be very dark or it can be like essentially white.
Yep.
It can have a very scabrous type, almost reticulate. Actually, there are reticulations occurring in the species from time to time. to something that's just white, and the scabers are completely white too. Yeah, very bizarre. Certainly, if you're in doubt, we should collect these and test them for something other than ITS, just to make sure. But this is becoming too much of a project. But yeah, that's what we're calling these at this time, albellum.
Yeah.
And
This one down here, Leccinum. Oh no, albellum. This one.
I mean, I would not call this Leccinum in a strict sense, but yeah, whatever name you choose, that's fine by me. I think it's closer to Hydnellum than Leccinum.
Okay.
If you think about the morphology, at least.
All right.
Oh, yeah.
Yeah, I agree.
Something I haven't seen in a while, unfortunately.
I saw one or two this year. I forget where I saw them. There might have been one at Mont Alto.
Maybe they were at Mont Alto.
Yeah, I think there was one.
I don't know how many there are in this spot here, but there were a lot of them.
Yeah, when I find them, sometimes there's a whole lot of them, but I don't find them all that often.
Did you eat any of them?
Yes, I tried it for the first time and it was nice.
It was nice, yeah. I mean, they're not like, wow, but they're not terrible.
Yeah.
I've eaten them. They're all right.
So yeah, so I dried up a bunch. I picked 24, I think.
Wow, very nice.
Yeah.
Yeah, other than rubropunctum, I think the rest are pretty good. Even that aenigmaticum, which is still of unknown edibility, I'm pretty sure is just as edible as sublabripes and hortonii. Oh yeah, very nice.
I don't know what it is though.
Very nice.
Oh wait, yeah. What's this?
Yeah.
Frosty eye.
Well, maybe Boletus subluridellus.
No.
You don't think so?
No.
The color profile is a little bit off for that.
Yeah, the pores are yellow and the blues.
Pores are a little bit too yellow. Yeah.
Yeah. Wow. Where did you find this?
In the field, I think.
In upstate New York.
Yeah. Yep.
And you tasted this, and this was none bitter.
Yeah, I didn't note that, but I don't think it was bitter.
And there's blue bruise on the pores, it looks like.
Oh,
yeah.
If what I think this is, then a big congratulations. This is a very rare species, and I just found this.
There were 4 of them at least there.
With Nina at the Washington Crossing. Did you save any of this?
I'll have to check.
Please
I might have some in the fridge, actually.
Yeah, please save this.
All right, I'll look and see if I got it.
I think
I love the reticulation. It was immediately caught my eye. Kind of a bland cap, though.
I'll walk back Exudoporus frosty eye, but that stipe is the only thing I saw before the pore surface. So Igor, what is this?
I think it's a Bolletus. I think this is biliae.
Oh,
biliae. Oh.
Whoa.
Yeah. And I have a story about biliae that's personal. Nina and I went to this foray at Washington Crossing because Nina set it up with the people at the nature center, and there was a fair amount of people from the public sector there as well. And after the foray, I dragged Nina to my place at Washington Crossing, where I have collected, mushrooms over the last 20 years. And lo and behold, I found my first collection of Bolletus biliae in a spot that I've been visiting many times every year, and I've never seen it there before. Of all the places, I found it there after 20 years.
Yeah.
So, did you cut this? It's stained blue inside, right?
Yeah, I don't know why I don't have a photo of that. I have my knife out, so I must have done something, yeah. But I'll have to look on my phone. Maybe I have more photos.
That's what I think it is.
Okay.
The caps are a little bit red for that, but this is not frosty eye.
No.
This
Yeah,
is
the
not Frostii.
cap's not red enough for that, and the pore surface is red on frosty eye, right?
Yeah.
Yeah, the Frostii is really like deep red. This interesting color of red.
Yeah, I think I have one of those. Some of those. If I,
and it's not just reticulate; it's fluted. So there are these deep,
Mm-hmm.
recesses or lacunae in the stipe, and it's almost like Rosellii, same kind of
Mm-hmm.
texture
Yeah,
of the stipe.
shaggy, or
Shaggy
yeah.
reticulate. Yeah, I see you have all sorts of bullets out there. So Viola, please save as soon as you can. Just throw it in the dryer.
all right, I'll look right after we get off. Yeah, here we go. Frosty.
Yeah. And let's not forget that Frosty has, red pores, obviously. So,
Yeah.
Yeah, my other ID was Boletus Pecii, but I don't think it's that. But Pecii should be bitter. So if you can maybe try it as well
Okay.
for bitterness. Biliae is mild. Pecii, Boletus Pecii should be bitter like all the Boletus. Yeah, I mean, there's no question. This is not that.
Yeah, this is Frosty Eye, this
Yeah,
one.
this is Frosty. Yeah.
Yeah, which was beautiful to see. It's funny, I was with a friend that I grew up with. She was back from California, and she saw this before I did, and said to her brother, like, "Oh, look, that's an apple mushroom." I said, "Oh, I think a candy apple is a common name for it, actually."
Right. So yeah, the Billy proposal, like I said, I think it should have more yellow in the stipe, in the upper part of the stipe. But other than that, it's a relatively good match. At least I cannot think of anything else other than Billy A. Uh, what's that one? You're on the
This
left.
one or
Yeah.
left? Yeah, I don't know. You tell me. When did I find it? Oh, today. Okay.
Okay.
A little bit of sterile material around the edge, it looks like.
Yeah.
Darker at the base.
No staining. Any staining?
Yeah, I think I know what this is. I mean, I
Oh,
suspect, but once I saw the cap, it was a little bit more straightforward. I think this is Pulchroboletus sclerotiorum.
Yellow
Hyphae.
hyphae down here.
That's yeah, and these will be forming sclerotia outside, on the leaves and then, on the soil maybe. Um,
Oh yeah, this one I was walking along and something had kicked it free. It was just lying on its side
right.
intact.
So it's either sclerotiorum or pseudosclerotiorum, and they are very similar looking. So you just propose one of them. We found pseudosclerotiorum in Washington Crossing because that's where the holotype actually is going to be from. Holotype collection. Typically, sclerotiorum has a narrow band of cap tissue on the rim as a defining feature. I don't see it here so much. So maybe this is the other one.
Okay.
By the way, Pulchroboletus sclerotiorum, that name will be retired and replaced with Bicoloroides, because Smith described that species from Michigan in his book in 1971, and we sequenced the type. So Bicoloroides is going to be a nom-nov.
so are all Smith collections from Michigan? Well, Smith and Thierce collections from Michigan. If they have a sequence, are there a lot with a type?
Well, yeah, I mean they have type collections for most of their stuff at Michigan from Michigan, and I think this is the same thing. Pulchra Bolletus, Sclerotiorum, pseudosclerotiorum. It's probably up north, so maybe it's pseudo.
This one's here in Chatham. This was today near
Yeah. So
near me.
right.
You think this is the same thing?
Yeah, this is the same thing.
Okay. Yeah, actually, I did think it was the same thing too, actually.
So yeah, Brandon, all those type specimens, they're type collections. And
Well, I more meant Smith and Thiers's collections from Michigan and the books that they wrote where they describe these species.
Yeah.
You said there are types for at least Pulchra Boletus. Do a lot of those, if I find a Smith and Thierce species, can I be confident that maybe there's a type and it's worth actually getting sequenced?
Yeah. If it's a species that was described by Smith and Thiers, all the boletes there have been sequenced by Dentinger and Damnauer. And I think Steve Russo is going after the types of all the other mushrooms at Mitch. And if you browse the accessions of boletes in GenBank, those Michigan collections are going to have MICH in the name, and usually the code is going to start like 10063. And when you click on the accession itself and look in the metadata, you'll see that it's a sequence by Dentinger and Damnauer. So it's the type from Smith, and it's going to say so in there as well. So yeah, if it's a Smith and Thier species in Boletaceae, you want to get it sequenced because chances are you can match it to the type. That's correct. And it's in the book.
Thank you.
Is anyone...
go to that Syzygospora. Syzyga, yeah, there we go.
Yeah, there was a fair bit of this. This was up in the Adirondacks on Saturday. So
just on the cap, or was there any on the base as well?
I don't recall seeing any on the base. I don't know why I only have one picture. Seems like I must have taken more, but
how did you know this Zygospora is something that's well known? Is it a parasite?
Yeah, it's particularly on this Colibia.
Dryophilus.
Yeah, which was everywhere. That Colibia was everywhere. I must have a photo of it. Yeah, here it is. Yeah, here's the...
okay, so the Gymnopus-like thing. Okay.
Yeah, all right, there it is there. Yeah.
Oh, okay. So again, we have a parasite that grows specifically on a given species.
Yeah.
Highly speciated.
Yeah.
Obligatory. Yeah, I've never seen these on Gymnopus dryophilus.
Oddly, I've only seen them in Michigan, actually. I haven't seen them in South Jersey, and we get Gymnopus dryophilus all the time, right?
Yeah, we get it all the time. It's very common. You should import these observations live into a Mo, now that it's so easy to do so.
Yeah, but last time I did that, it said everything was in Massachusetts somewhere. It didn't get the address. Maybe I did something wrong on the import. The location was all screwed up.
Were you doing this from your phone or from the computer?
The computer, the import. I put them all into iNet, and then I went and I imported like 10 at a time, and it said they were all in Goshen, Mass, or some crazy place.
Well, did you report this to the MO administrators?
No, not yet.
Yeah, because this is something that should be done because they're actually chasing all the bugs.
All right. Well, yeah, I wanted to talk to you about it because I went to try to edit my observation, and it seemed like I was editing the definition of Gaussian mass, and I'm like, I don't think I want to be doing that.
No, you
So
just want to replace one location name with the other.
yeah,
You want to edit and you want to just erase and write another one in.
yeah. So so I need help with that. But anyway, so this is my first time successfully digging up one of these things, so I'll show it to
Yes.
everybody.
Phylloporus, is it?
The
Phylloporus, is it?
yeah, Tylopilus ophidiosoides or
Of the glossoides or something
yeah,
like that,
something
right?
like that.
So this is
Like
growing on a another
truffle,
mushroom, truffle. Okay.
truffle. Yeah, here you can see it better.
So here we go with another story. It's another parasite and another highly obligatory relationship with a specific host.
Yeah. So I was lucky. It wasn't buried too deeply.
Yeah, and these Ophioglossaides, the rhizomorphs are much more fragile, so this is good work.
Yeah. Yeah. So I saved it because I had never done it before, so I've got it.
Yeah, very nice.
So that
Very
was
nice.
fun. Ash tree bolete.
Yep.
Found several of those.
I didn't see them anymore here because all the ash is gone.
Yeah, was at the base of a healthy ash about 10 inches in diameter. Very close to the base. There were,
Mhm.
I don't know, 6 or 7 fruit bodies, 3 or 4 right in this one spot. I
Yeah.
just love the pore
Oh
structure.
yeah, the pores are great. You know, they're compound, they are bolletinoid, and you can appreciate the honeycomb structure, a layer after a layer after a layer of these almost like lamellate-like pores,
Mm,
because you can follow the lines radiating. It almost looks like a gill that's just branching and branching and forming these honeycomb structures. Look at that. I mean, how many layers of that stuff that we have inside? Just fantastic.
yeah.
That's a beautiful photo.
Thank you. Yeah, so that was fun.
Yeah, this species should be evaluated for, being a red book, species,
Oh, red listed, yeah.
yeah, whatever you're right. Yeah, you know what I'm talking about.
Yeah. I see
Something
you on red list, yeah. All right, is
the
Dave
red
still
list, yeah.
here? Does he know this? It was interesting, very much an odor about it. I instantly thought smoke. It's funny. I don't usually, um,
Trichoma odorum.
yeah, no. Then I had Dave smell it and he said immediately soap. So somebody came in and said saponaceum.
Yeah, that's a dirty name. Yeah,
I don't know. Does Dave know this?
Dave is probably asleep.
Okay.
That's what happens when we have long nights at Taxonomy Tuesday. Yeah, it's funny that we never found this tricholoma in New Jersey
Yeah.
that smell like soap. Of all the tricholomas we've seen, including some of the rare ones like Grave, we've never found saponaceum.
Yeah. There are a
And
bunch of them.
odorum has a much different smell, right?
Yeah,
Mothballs.
yeah, yeah,
Yeah.
and it's more kind of green in appearance, I think,
Yeah.
and it's smaller. It's not a very large mushroom. So yeah, you got the Rubra. I'm not gonna say anything about that. That's what I would call it. Although there's a lookalike, and then you have a malaria pallida. That's also a good ID. And yeah, we have all the other ones that we discussed, I think, with you via email.
Yeah.
So
That's-
Macellenta, Megaloma, right? Cool plant, yeah.
I'm not positive about this one. Maybe Brandonus does this better. Let's see. There were two bigger ones. These are the two bigger ones. That's the base.
Look at those tightly packed gills.
I wouldn't argue with it,
Okay.
but I know enough to just not be confident about these things.
All right. Yeah. I certainly am not confident about them.
I mean, when
But-
you, sorry to interrupt, when you post these things, Lyle, does INET immediately involve AI to propose, suggest names to you, or this is something which is optional?
Yeah. Well, you can go and immediately put in what you think it is, but if you take your time, it's going to suggest things to you.
And have you been finding that functionality helpful?
Yeah. I think it's often quite good. In fact, on that one that you gave me the name on the Whitey's bolete or whatever, that was one of the options that it said.
Which bolete?
Where is it?
Aureoboletus.
Was it this one? Yeah.
Oh,
No.
Did it give you an
It
option?
didn't take my name. Did it not take my name? I thought we put a name on that.
I thought I saw you put a name on that.
Yeah. Maybe it was this one.
Oh, you
Oh,
might
that one
have
was
to.
biliae. That one, the other one was biliae. This one. Yeah, Whitey's bolete.
Oh,
Pulchroboletus.
Whitey's bolete. Okay.
Yeah, I remember that just because I know somebody that we used to call Whitey. Yeah. So that was one of its suggestions, maybe the second or third suggestion. I can't remember what some of the others were, but I remember.
By the way, for this bolete, Igor, you recommend not to eat it. I realize I have ate that before. Last time when I cook it, I turned carrot
Orange.
color. Yeah, orange.
Yeah, orange or like reddish orange. Yeah, that's what they do. I'm not sure if both of them do it, like both sclerotiorum and pseudo sclerotiorum, but one of them does that. Yeah, the reason I do not recommend eating Pulchra is because generally the edibility of that genus is unknown, and there's one Pulchra by the name of Miniata elevaceus that apparently is gastrointestinally toxic.
Yeah, that one's bad.
Which is surprising. And Smithii, its analog, its next of kin from the West Coast, which looks almost identical, has been implicated in poisonings out in the West. So that's the only reason why I said, well, Pulchra I'm not going to risk it. But it's possible that the genus consists both of edible and toxic species. But you said you ate this and then you were okay, right?
Without knowing it. Remember last time I shared a photo that because I saw the orange color, but apparently I shared a raw mushroom photo. Later I realized that's what I ate, but nothing happened. Just
nothing happened. Good. Well, now we know that it's edible. except the question is, did you eat sclerotiorum or pseudosclerotiorum?
That's right. Yeah.
Well, it would have to be pseudo, right? Because the other one is known to be, unsettling.
No. I said Miniata elevaceus is known to be unsettling, but I didn't say anything about sclerotiorum or pseudosclerotiorum.
Yeah.
Oh, sorry. Yep, misunderstood.
All right, here's this one that you think might be biliae. Yeah, so I didn't give that option, but Boletus brunneus was one of the options.
No, that's not that.
Yeah, so
It's not
the more rare it is, the less accurate the.
You can maybe it's going to accept it as Boletus biliae because it hasn't been officially transferred into Boletus, but that's where the genes place it. Yeah, there you go.
Lila, you were 100% correct. The less commonly observed a thing is, the less reliable the AI is going to be.
Yeah.
You know, uh, I think what was the example? Bolletus purpurea rebellis, right? I know exactly what that thing is, and I checked the AI, and it wasn't on the top 10 for any of my photos. Like it wasn't in the list, and it's such an indistinguishable mushroom. So it's all about feeding the AI algorithm.
Yeah, has to learn it.
Right. Yeah.
Did I
So, but yeah, please do preserve it because, like I said, there's a chance it might be something else.
okay.
Um, and if you are brave enough to taste it, go ahead and do that too because that's going to be even more helpful early on.
All right. Well, I'll go see if I know I threw 3 or 4 things in the fridge, so
Yeah,
might be
please,
might be
let
one
this
of
be
them.
one of them. Yeah.
Okay, I hope it's one of them. We'll see. What's this one here? Oh, this is from a week ago. Where was that? Locally here somewhere.
Huh. Well, would I be off to just say subluridellus?
The caps are brown.
Okay.
Yeah, red up top or blue up top, kind of olivey in the middle and reddish down below.
Yeah. There's a lot of staining, and I would love, to have seen the original stipe surface before the staining, where they had
What it looked like.
brown squamules, because this might be, although would be probably no, probably not out of realm of possibilities because that species is known up north, up to Canada. This could be Neoboletus brunneosquamulosus, and I think that's what it is.
Yeah, look at that.
Congratulations, Lila, if that's what it is. That's another uncommon species. I'm going to put the name in.
Okay.
I don't know if the AI is going to accept this, but I can guarantee you that MO will.
All right, what did I say about it? Yeah, didn't smell great. Very firm.
Yeah, just to remind everyone that stipes are probably the most discerning morphological or anatomical structure in boletes, and that would be responsible for the majority of the identifications, followed by the caps.
The stipes are the most important.
The stipes are more important, yeah.
Okay. All right, well, good.
There is again a possibility of the chameleon, but I would rather propose this name and be wrong about it. And
Okay.
it's just that's what I think it is based on the overall morphology. But I've been fooled about the chameleon more than once. It's such a shame that we have to live with subluridellus because that's such a boring
Name.
boring and unimaginative and undescriptive name. And that subluridellus is not subluridellus, probably. Or maybe it is. I don't know. I'll have to see. Caps are too dark. They're reddish brown. I think this is that aenigmaticum, maybe.
Oh, with um the white stuff here on the top.
Yeah. Yeah, it's possible. I'm not sure how far that is distributed. I think I said Connecticut.
Yeah.
So, and there were no collections from New York, but I'm a little bit suspicious about this being sublabripes.
Yeah. Is
And
this
not the best shape, but the colors are too dark, I think. They're too reddish brown as opposed to caramel brown for
okay?
sublabripes.
Would you? I couldn't really. I don't think I could decide. This sort of looks like it wants to be reticulate, but
Yeah, well, I don't know, Loyola.
all right.
Yeah, so
There's inside.
it's yeah. Is that bluing or this is just like an
I,
aberration?
I think there was just a tiny bit of bluing here.
Just to let you know that sublabripes can blue. I have an example of that. That's
Okay.
genetically is similar to something from New Jersey that is not did not blue at all. So bluing can occur in Hemilecccinum, in some Hemilecccinum. I don't think I've seen it in aenigmaticum, but it doesn't mean it cannot happen. For example, Aureoboletus vinacepes, the description says that it blues in this type. I haven't seen a collection in New Jersey of that species that blued ever. Yeah. Well, now the mushroom on the left with a lot of redness in its type, that looks more like rubropunctum, actually. Did you smell this one?
Yeah, I didn't get the ashtray. Yeah.
Yeah, that's true. But
Yeah.
it doesn't always manifest itself. I mean, I've
Mhm.
smelled clearly rubropunctum, and it didn't have that odor. So not every collection of rubropunctum is going to smell like an ashtray.
Okay.
Just an anecdote. We found a bunch of rubra punctum. That one I think I know at Shark River Park, and I'd say 1 in 3 had the actual odor.
Huh.
The one that I collected in the field had it. Everyone smelled it. Maybe it goes away once it hits the ID table, but we found a couple more, and the other ones didn't. So
Yeah,
Mhm.
well, it's believable. I mean, I experienced the same thing, and there's nothing wrong with my nose. After all, I can still smell things like Amanita mauricii and Amanita submaculata with that fine, minty toothpaste, odor, and others cannot smell it. At the same time, I couldn't smell anything about Pseudomyrurlius curticii. People said how terrible and disgusting this was, and I'm like, guys, what are you talking about? This doesn't smell like anything at all. It has no odor. And that was at NEMF, and people saying, are you kidding me? I mean, this thing reeks.
yeah,
But I don't
yeah,
think so.
but even this past weekend, right? There are 4 of us standing around, looking at the same mushroom and with the same description in mind. Like this is supposed to smell like this, and one person smells it, or 3 people smell it, and one doesn't, or the other 3 don't. You know what I mean? So it's not everyone is picking up that odor.
Yep. Well, that's believable. I mean, I would believe it more with sense and odors as opposed to taste. But some people cannot taste bitterness, or their ability to taste bitterness is reduced, and they say, "Well, it was just slightly bitter." And I'm like, "Are you kidding me? This is plumbeoviolaceus. I mean, it's going to rip your tongue out.
Right,
You're going to
right.
taste that for like hours."
And we were smelling Volimus comes to mind. Me, Nina, uh, I don't know the other person's name, and Chris. We were all smelling a Volimus, and I smell it. I'm like, oh yeah, that smells like fish.
Mm-hmm.
And I pass it to the next person. They're like, yeah, fish. The next person, I don't get that. It smells like soil, just soil.
Hmm.
Like, you know, Yeah, soil at low tide, maybe, but to my nose, smells fishy.
Yeah, it always smells fishy to me after it dries. Okay.